So Santa, why don't you call the meeting to order?
I will do. Good morning, everyone. I'd like to call the 155th meeting of the National Advisory Eye Council to order. I'll turn the meeting over to Dr. Ann Schaffner for introductions and some housekeeping information.
Okay, so Mike, I see that you've started the recording. That's great. And I'll serve as a reminder to everyone here that our open session of council is being broadcast live and recorded. So please remember to mute your microphones when you're not speaking or asking a question so we can minimize the background noise. These are very challenging and unprecedented times. I'd like to think that as human beings, we're resilient. As people involved in many different aspects of science, we're creative. And as government servants, we are dedicated. So here we are at what I believe is our first virtual council meeting. I'll start by thanking all those present, our council members. We have 4 new members and Santa will introduce them later. NEI staff, other NIH, including scientific review officers and IRG chiefs from the Center for Scientific Review, and our advocates for vision research, including ARVO and NAVR, N-A-E-V-R, and many members of ophthalmology and optometry associations. I also include a welcome to others who might be watching this video cast of our open session, including members of the public. So there are just a few housekeeping details that I need to go over very briefly. I want to thank, first of all, the council members and for others who sent in their pre-conflict of interest forms for the closed session. It was important that we get those before we start discussion in that session. I'd also like to mention some tips and Zoom etiquette for the meeting panelists. So during a presentation and the discussion afterwards, unless you're a presenter or you're asking a question, please mute yourself and turn off your video. This serves 2 purposes. One, it will preserve bandwidth and allow for a better flow to the meeting. And it will also allow the interpreter's face tile to be up front and center. So those who need to see the interpreter can do that easily. They can also pin the interpreter's face tile by using the 3 small dots in the upper right-hand corner of their screen. If anyone wants to pin the speaker, they're free to do that as well. I think for the best flow for the meeting, it would be best to wait until the end of a presentation before anyone asks a question. So there's that raise hand option for panelists at the bottom of your screen, or if you open up the participant list, you can also raise your hand there. It brings all the raised hands to the top of the list so I can call you in turn. When I mention your name, you can unmute yourself, show your video if you want, ask your question, and then the presenter can answer that, and then just remember to remute yourself. Please state your name again before you ask your question. And that's mainly for accessibility purposes. For the attendees, there's a Q&A. So I can open that up as well. So after council members and panelists have had a chance to ask their questions, I can open up the questions in the Q&A and get those answered. So for the approval, we have 2 PARs being reissued and 2 RFAs and several concept clearances. So in order to make voting for those easy, I think what I'm going to do is use that raise hand icon. That way I don't have to search over 2 pages of face tiles to see who's got their hand physically raised. I can just do the count in the panelist column there on the right-hand side. So that's all I have to say in terms of some opening remarks, and I'd like to turn it back over now to Dr. Tuminia, the acting director of the NEI, for her opening remarks. So go ahead, Santa.
Thank you, Annie. So I'd like to add my thanks and welcome to all of you for joining us. We have members of NEI staff, members of NIH, council members, and we also have members attending from our advocacy and other stakeholder interest groups. So welcome all of you. I'd like to thank you for joining us to our first virtual meeting, and I'd like to ask you to just bear with us if we experience some bandwidth issues or other technology challenges. So first, I'd like to start by welcoming our newest council members. This is not exactly the warm welcome that we had in mind for you, but we're certainly glad that we're able to have the capability to work with you to conduct council business virtually. So first, I'd like to introduce Dr. Turete Borras, who's a professor in the Department of Ophthalmology at the University of North Carolina. Dr. Borras's experience is in trabecular meshwork, glaucoma, and the genes and cellular mechanisms that regulate intraocular pressure. Her group is also working on gene therapy approaches for the treatment of glaucoma. Next, Dr. James Coughlin, who's a senior scientist at Smith-Kettlewell Research Institute in San Francisco. His expertise is in the design and development of assistive technologies for the blind and visually impaired. His background is in physics, computer engineering, mathematical modeling, and neuroscience. Next, we have Dr. Tom Gardner, who is a clinician scientist and professor of ophthalmology and visual sciences at the Kellogg Eye Center, University of Michigan. He's also a professor of internal medicine, metabolism, endocrinology, and diabetes. His expertise is in the design and execution of clinical trials in retinal diseases, including diabetic retinopathy and macular edema. We have Dr. Renu Kalluru, who is a professor at the Department of Ophthalmology, Visual and Anatomical Sciences at Wayne State University School of Medicine. Dr. Kalluru's expertise is in diabetic retinopathy, specializing in metabolic memory and mitochondrial function. She's also interested in the role of epigenetics in the pathogenesis of diabetic retinopathy. So welcome to all of our new council members. I'd like to continue with some staff updates. I'd like to share, I'm sure all of you have a burning question of when will there be a new NEI director announced? So as most of you remember, Dr. Paul Sieving, our former director, retired from NIH last July to join the faculty of UC Davis and to create a new center for ocular regenerative therapy. So it's been almost a year now, and I'm sure many of you are wondering where we stand in our search. Well, despite COVID-19, we are actually still on target with our search. And the search has continued. Dr. Collins has been working very, very hard. And all I can say at the moment is that we do expect an announcement soon. Soon is relative, so you'll just have to bear with us and stay tuned. And while I know that this might seem like a long time, actually, this is moving at a very good pace. Normally, a search takes about 18 to 24 months, and that's without a pandemic. And in addition, Dr. Collins is actually conducting 5 searches, so 4 other searches besides the search for the NEI director. And he just announced one of the new director yesterday for one of the other institutes. So the searches really are moving along, and all I can say on that right now is to stay tuned for more information. Connected to that search is the search for a scientific director. So as many of you remember, Dr. Sheldon Miller, our former NEI scientific director, retired from NEI last March. So we placed the search for the scientific director on hold until after a new NEI director is selected. That way, the new director has the opportunity to forge his or her vision for the intramural program, along with the scientific director that he or she has chosen. So while we're waiting for the new director to be selected, Dr. David Schneeweiss continues to serve as our acting scientific director. We've had a number of departures since our January council. From the extramural program, you will all remember Dr. Jerry Wujek. He retired last month after 14 years in the NEI's Division of Extramural Research. He served as NEI's Resource Officer. He came to the NEI in 2006 and managed the grant portfolios for small business and for the BRAIN Initiative. And for those of you who joined us, who were with us last year in our last councils, you'll remember that Jerry gave a fabulous presentation on some of the successes in the SBIR and STTR portfolios. From the intramural program, Dr. Y. Wang, who is a senior investigator and chief of the section on neuron-glia interactions in retinal diseases, he left the NEI in February to become vice president for retinal diseases at the Janssen Pharmaceutical Companies of Johnson & Johnson. Dr. Wang is a clinician scientist, and while he was at the NEI, he was highly productive, and his team worked on the role of innate immune system in healthy and diseased retina. He's especially well known for elucidating the basic physiological properties of retinal microglia, including how they can contribute to the degeneration of photoreceptors. We wish Dr. Wang much success in his new position, and Dr. Wujek much happiness in his retirement, although we will be calling on him for some assistance as we go through a transition period. So with those departures come arrivals. And I'd like to welcome Dr. Pak Lee, who recently joined the Division of Extramural Science Programs and has taken over managing the small business grant portfolio. And he will also serve as the NEI's Resources Officer, the position that Jerry Wujek vacated. Dr. Lee comes to us from the Center for Scientific Review, where he was a referral officer in the Division of Receipt and Referral. He received his PhD in neuroscience from Kent State University and completed postdoctoral training at the Uniformed Services University of Health Sciences. He came to NIH as a fellow in NINDS in 2001 and joined CSR as a scientific review administrator intern in 2003. He became a scientific review officer in 2005, and he also completed a detail at the NEI's Division of Extramural Research in 2012. So welcome to PAC. Next, I'd like to introduce Dr. Gordienko, who also recently joined the Division of Extramural Science Programs. Dr. Gordienko comes to us from the Center for Scientific Review. You might detect a theme here, where she's been a scientific review officer since 2009. She earned a PhD in biophysics from the Academy of Sciences of Uzbekistan and completed 2 years of postdoctoral training in the Institute of Bioorganic Chemistry in Uzbekistan. She came to the NEI as a visiting fellow in 2002 and worked in the laboratory of Dr. Ignacio Rodriguez in the Mechanisms of Retinal Diseases section. She continued to play a role as a senior biologist in several NEI laboratories from 2006 to 2009. Lastly, I just wanted to update you on the search for a new director for the Division of Extramural Activities. I just wanted to let you know that we have selected a new director, and we look forward to having our selectee in place soon and meeting with council members as soon as possible. However, I can't say much else about the selectee because we have to wait for HR to complete its many processes. But at this time, I'd like to take the time to thank Dr. Annie Schaffner for her dedication and commitment to serving as acting director of this division since last January of 2019. Annie's done a fabulous job, and I know she very much enjoys interacting with all of the council members on our business, so I just want to thank her again, formally and officially at our council meeting. So thank you, Annie. In some other news in our intramural program, I just wanted to bring to your attention that Dr. Kapil Bharti, a senior investigator in chief of the Ocular and Stem Cell Translational Section, has received IND approval for his novel stem cell derived RPE replacement phase 1 clinical trial. It's a patient-derived stem cell therapy to replace and repair dying cells in the retina to treat geographic atrophy, the advanced dry form of AMD. And clinical trial began enrollment in the spring, and currently there are 3 patients enrolled in that trial. The trial calls for an enrollment of 12 individuals. So with that, I'd just like to give you a brief overview of today's presentations. We'll be hearing from Ms. Karen Colbert, Budget Officer and Head of the Financial Management Branch. Karen will be discussing fiscal year 2020 appropriations and NEI budgets. Dr. Russ Van Gelder will share with us a debrief of the Council of Councils meeting on which he's a representative. For those of you who are new to NIH and its many committees and meetings, the NIH director, so Dr. Collins, he has several federal advisory committees that are usually comprised of members who are outside of the NIH and have the required expertise to advise him on a variety of topics. The Council of Councils is made up of approximately 30 members of other institute and center council members, and they serve to advise him on a number of activities. In this case, it's related to the policies and activities of the Division of Program Coordination, Planning, and Strategic Initiatives, otherwise known as DPCIPSI. And so Dr. Van Gelder will share with us his experiences on the council for that Council of Councils for their last meeting. We'll also hear from Dr. Sheffy Gordon, who's Director of the Office of Program Planning and Analysis, and he'll update you on the activities of our strategic planning process, and it's called 2020 Vision for the Future. Just one thing I'd like to bring to everyone's attention is that the strategic planning process is the strategic planning process. There is nothing different about it, whether we have an acting director in place or we have a permanent director in place. And so we don't want anyone to get the impression that we are putting out a strategic plan right before a new director comes so that she or he wouldn't be able to implement his or her own vision. In point of fact, the strategic planning process actually has two phases. The first phase is information gathering. That's when the NEI talks to the community and our stakeholders to see what's on their minds, what are the gaps and opportunities that they perceive. And we impanel panels and working groups to give us ideas and make some recommendations. And so we're still in the information gathering phase, and we will not be issuing a plan before a new director is brought on board. The information gathering phase will help the new director forge his or her vision and implement that vision for the NEI. So I just wanted to make sure that everyone is clear on that. We know there have been some concerns from the community, but we will be waiting for a new director before we issue the strategic plan. Next, we'll be hearing from Dr. Steve Becker from the Office of Regenerative Medicine. He'll provide updates on audacious goals and regenerative medicine activities, which are part of the new Office of Regenerative Medicine. He'll also bring up a concept clearance for the development of an AMD Integrative Biology Initiative. And as Annie mentioned earlier, we'll also be hearing about other concept clearances from Drs. Tom Greenwell and Martha Flanders. We'll also get an update on the Anterior Segment Initiative, also known as ASI. It's a new research program aimed at tackling the difficult to treat diseases of the anterior segment, and it's meant to complement our AGI initiative, which is focused on the posterior segment of the eye. At our last council meeting, we mentioned that we had put out a request for information in the fall on ASI, and that RFI closed in January, so we'll be hearing some of what came out of that. We received more than 50 responses to that RFI, and that represented about 200 stakeholders. Some topics of interest that rose to our attention from that centered on inflammation, ocular pain, immunology, and the microbiome. Staff have been having meetings, and we've discussing various ways to tackle the different topics. And today you'll be hearing from Drs. Sangeeta Bhargava and George McKee on inflammation. So now I'd like to switch to some of the broader topics that have the NIH's attention. And I also want to point out different places that you can go for more information, as we'll just lightly be touching on some of these topics today. So yesterday, the NIH released new guidelines to bolster its policies and practices to address and prevent sexual harassment. So there are new communication channels to inform the agency of instances of harassment related to NIH-funded research. And yesterday in an editorial in Science magazine, Dr. Collins announced a change that will hold grantee institutions and investigators accountable for this misconduct. So I encourage you to look at that, as well as to look at the new guide notice that's also out that discusses what types of situations are covered under these new guidelines and what the roles of the grantee institutions will be in these instances. Now, as we talk about sexual harassment, we know that there is a lot of societal unrest. I feel like now we're at a place in our country where there's much more societal awareness and societal responsibility. Some of that comes obviously from COVID-19 and worrying about not only protecting ourselves, but the people around us. But, you know, it's a number, the pandemic brings its own challenges, and then the societal unrest brings some others. And at the NEI, we plan to meet all of these challenges with strength and compassion. We're going to take this opportunity to recommit ourselves to the goal of supporting research outcomes that benefit all of us. And with the death of George Floyd and others, the NIH and the NEI are committed to using every strategy that we can to enable and incentivize institutions to develop structures and policies and cultures that are not only diverse, but are also inclusive. It's invited to the dance, but you also need to dance. And so we want a safe environment for everyone. We want to be supportive of all of our community members, including those from groups that have historically been and continue to be subject to discrimination and health disparities. We at NIH actually have a critical role to play here, and by ensuring that we can foster a culture of inclusion, equity, and respect for one another, and by ensuring that the research we support is part of the solution rather than part of the problem. And this really comes to our attention, especially in health disparities where COVID-19 is concerned. And we know that while we can be overwhelmed at times with challenges, at NIH, we tend to rise to the occasion of a challenge. And so you might wonder, what have we been doing while this pandemic has been going on? Well, NIH has mounted an incredible response to COVID-19. There have been many planning strategy meetings to address the pandemic, everything from the basic biology of the SARS-CoV-2 virus and its proteins, as well, everybody has heard about diagnostics and testing. And all of the institutes, including the NEI, have been involved in planning processes to think about the types of things that NIH could be involved in should there be an appropriations for work like that. And there are many, many different activities. Obviously, we can't go into them today, but we really wanted to be ready to hit the ground running. And actually, the federal government did give NIH an appropriation in the amount of $3.6 billion to address COVID-19. Most of that money is with a strong emphasis on diagnostics. So NIH has developed several cross-cutting and public-private initiatives to address COVID-19. One of them was announced on April 17th. That is called ACTIVE. And ACTIVE is short for Accelerating COVID-19 Therapeutics. I've just lost. Therapeutic interventions and vaccines. And basically, this is to develop a coordinated effort among both the public and the private milieus. We're in the attempts to develop not only diagnostics, but also treatment and vaccine options. So this is coordinated by the Foundation for NIH, and it brings together about 7 different partners from government. There are 18 different pharmaceutical companies involved and several nonprofit organizations. And this group the goal is to prioritize and leverage candidates and to accelerate vaccine production. They also would like to accelerate clinical trials by utilizing existing networks. So everything is geared towards coordination, leveraging what's already available, and prioritization. There's also a lot of time spent on defining and developing master protocols so that people are using the same types of protocols. And there are 4 working groups that are involved in this program that include over 100 people. The different working groups are the preclinical working group, clinical therapeutics working group, clinical trial capacity working group, and vaccine development working group. And this, as I said, is to standardize and share preclinical evaluation methods, prioritize and accelerate clinical evaluation of candidates with near-term potential, and to maximize our clinical trial capacity and effectiveness. There's also a second program, which some of you may have heard, called RADx, which is short for the Rapid Acceleration of Diagnostics. There are actually four currently different programs in RADx. The first is RADx Tech. That's a $500 million program, which is a 3-phase challenge to find the best candidates for at-home or point-of-care diagnostics. And it's to develop rapid and easy to use testing technologies for SARS-CoV-2. Now, this has a phased development. So there was a national call for innovative technologies, and one didn't have to be very far along in their diagnostics to apply in this phase. And so if they were chosen, they passed to the next phase, which is kind of phase 0. And if you are fans of the Shark Tank, this is likened to a Shark Tank approach. It's a selection process. And if you pass the Shark Tank, you move on to the validation and review phase. And then if you get through that, then you go through phase 2, which is clinical testing and regulatory approval and scaling up. And so to date, that program has received 2,200 applications. 58 of them have actually made it through the Shark Tank, and 9 have made it to phase 1. So we'll be watching that space very closely. Following on that is the RADx ATP program, which is the Advanced Testing Program. That's a $230 million program, which are for those technologies that are past the Shark Tank, but not quite ready for scale up. So that is looking to be like contract mechanisms, and the awards should be made by the end of this fiscal year. The third is called RADx Rad for RADx Radical. That's a $200 million program to develop and advance novel, non-traditional approaches for, or new applications of existing approaches for diagnostic testing. And they look to use a range of mechanisms, including cooperative agreements, SBIRs, R01s, to support awards that would be made by the end of 2020. And then the final one is called RADx Up, and that is short for RADx Underserved Populations. That's also a $500 million program, and those surround interlinked community-based demonstration projects that are focused on implementation strategies to enable and enhance testing of COVID-19 in underserved and vulnerable populations. So populations that are not traditionally at the forefront when we're thinking about these kinds of scientific activities. This is very focused, and it has the support of almost all of the institutes. There's a phase 1 that has data coordination and collection and collaborative networks. And then there's a phase 2, which are expansion of the phase 1 projects. We can't really tell you too much. The NOCs and the FOAs were supposed to be out this week. Dr. Collins did mention these, and there were some explanations of the programs a little bit deeper at yesterday's ACD meeting. So that's Dr. Collins's advisory council to the director meeting. And if you're interested, those slides will be online, so you can see for yourself, but I would encourage everyone to pay attention, because NEI is very involved in all of these COVID-19 activities. We have a number of NEI staff, including Dr. Sangeeta Bhargava, Dr. Don Everett, Jimmy Lee, James Gao, Sherry Wiggs, and Carrie Getz are also all involved in these programs. So there'll be more to come, but we encourage you to pay attention to the NOCs and the FOAs. We also have an intramural response to COVID-19. Like everyone else, our intramural community is mostly teleworking, and the only staff allowed on site are clinical staff, as well as those who are responsible for maintenance and animal care. But there's also been an opportunity through NIAID for the intramural program to participate in COVID-19 research. And we have several intramural investigators who have proposed projects in COVID-19, and we are waiting to hear the results of those applications. As for what happens with our extramural community and our normal vision-related grants that are unrelated to COVID-19, we're all very concerned, just as all of you are, about how all of our labs have been forced to close down and experiments are on hold. So we are supporting the extramural community as best we can right now by exercising maximum flexibilities within any allowable limits. So if you have questions about grants, please contact your program officers. We are waiting to hear if we will get additional guidance from downtown regarding what to do to quote unquote make grants whole. At this time, we don't have any information to impart, but we are advising researchers to apply for no-cost extensions when appropriate and to monitor the guide notices for funding opportunities that might address any needs the extramural community will have. So with that, I'd like to end my opening remarks and turn it back over to Annie or any questions that you might have.
So are there any panelists who have a question for Santa regarding her remarks? All right, I don't see any hands, so up, Mary Elizabeth has one, so.
Yes, thank you, Santa and Annie. My question has to do with, is there any concern that the money used for COVID-19 will in any way compromise the amount of money for other research being done in eyes, in ophthalmology or eye research?
So at this point, no, those were separate appropriations and shouldn't impact our current appropriation or future appropriation. But as you know, everything is evolving. So we're taking mostly a wait and see kind of attitude.
Okay, I see no other questions. So I think I'll just carry on here. There's just a few administrative details that need to be taken care of. The first one is the January minutes. So are there any changes or corrections? Anyone who read them, let me know. You can raise your hand if you saw something that I need to edit. If not, then what I'd like to do is have council members who approve of the January minutes just use that raise your hand sign, and I can just do a visual count, 8, that would be a quorum. Is there anyone who does not approve? Okay, it looks like there's a consensus for approval, so I thank you for that. I posted on the ECB, I have this ongoing table of all of the NEI sponsored PARs and RFAs. So PAR is an acronym for Program Announcements Reviewed in the Institute. And RFA is Request for Applications. And the NIH has said that ICs must present in an open session of some sort. It can be council, it can be a public meeting, when they are issuing or reissuing a PAR or RFA. So especially the ones, notably the ones that we sponsor as an institute. So there are 2 PARs that we are going to reissue. And one is the R24, that's our translational research program for the development of visual disorder therapies. And the other is our K12, that's the Institutional Mentored Physician Scientist Award. So what I need from you is approval for us to reissue those 2 PARs. One expired in, I believe, April, the other will expire today. And so the next the next reapplication deadline is not until next spring, but it takes a while to get them through the system. The program officer on both of those PARs is Neeraj Agarwal. So Neeraj is a panelist. If there is anyone who has a question for Neeraj on either the R24 or the K-12, you can ask it now. You can raise your hand. Russ has a question. Russ, unmute yourself, or I can do that, and ask your question. And then Neeraj, if you could unmute yourself to answer. So go ahead, Russ.
Great, thanks, Amy. So on the K-12 program, my understanding was that we had, it was being de-emphasized, and that the emphasis on the K programs was going more towards the individual investigator awards because of a much higher success rate in conversion to R01. I'm just curious to what extent, if this is reauthorized, it will be emphasized or de-emphasized within the portfolio.
So Neeraj, can you answer that question?
Sure, I could. First of all, K-12 is coming to an end when we are reworking on that. So the announcement is coming back. So it's not going away. That's number one. And number two, as you just said, that the individual Ks have better success rate of R01. So that doesn't mean that we are getting away with K-12. K-12 is here and it will come back. So I don't see any reasons to worry about it, if I answered that correctly. Mike?
Yes, Russ, so the way that it stands right now, you know, the majority of our K money goes to slots in these K-12 grants. And in response to that, we're slowly migrating our K-12 slots into individual K awards. As a result of that, these findings from the National Academy. So we're being very careful about this. We're trying not to pull the rug out from underneath departments who are, you know, depending on these. And we certainly understand the need for departments to have money to attract a really good applicant. So we'll continue to move things towards the individual grants. But it'll be a slow transition and something that we hope won't upset departments depending on these monies.
Perfect, thank you, Mike.
Okay, I don't see any other questions. So what I need now is for all council members in favor of reissuing the FOA for the R24 and K-12 to again use the raise your hand button. Okay, thank you very much. That looks like a consensus. So thank you. And later in the open session, we're going to hear about concept clearance for 2 of our RFAs, but we'll do that later. So now we can go on to our speakers. And our first speaker is going to be Ms. Karen Colbert. She's the NEI's budget officer, and she will give us an update on the budget. Always interesting information, especially in these times.
Absolutely. Good morning, everyone.
Slide. Budget update. National Advisory Eye Council, June 12, 2020.
And so please let me know if any point you have trouble seeing the slides or if they're not advancing properly. And can you hear me okay? Okay, fantastic. So again, I am Karen Colbert, the NEI Budget Officer from the Office of Administrative Management, Financial Management Branch. And again, I'm pleased to be here with you this morning to share our budget update. I'll let you know that I will describe the content of many of my slides in order to assist with the sign language interpretation and the captioning.
Slide. NIH and NEI competing RPG success rate. FY 2013 through FY 2019. Table, NIH versus NEI for each year. Listed are applications reviewed, applications funded, then the success rate, which is highlighted. 2013, NIH, 49,581. 8,310, 17%. NEI, 1,128, 267, 24%. 2014, NIH, 51,073, 9,241, 18%. NEI, 1,085, 290, 27%. 2015, NIH, 52,190, 9,540, 18%. NEI, 1,278, 273, 21%. 2016, NIH, 54,220, 10,372, 19%. NEI, 1,294, 332, 26%. 2017, NIH, 54,005, 10,123, 19%. NEI, 1,222, 304, 25%. 2018, NIH, 54,834, 11,071, 20%. NEI, 1,159, 310, 27%. 2019, NIH, 54,903, 11,035, 20%. NEI, 1,179, 335, 28%.
So before I start to discuss where we are with 2020, I wanted to share an update to the NIH and NEI success rates that were not available when we were together in January. And so what we have here is a multicolored table that shows the history of NIH and NEI success rates from 2013 through 2019. 2019 is the latest fully completed fiscal year. For both NIH and NEI, the columns show the total number of applications reviewed for each year, the total number of applications funded for each year, and the calculated success rate for each year. Success rate data is captured by the NIH Office of Extramural Research, and we regularly share the success rate data because frankly, it's a measure of accomplishment that Congress looks at. Perhaps in the future, Congress will focus, their attention on other measures, and I'm sure they do to some extent. But for now, success rate is what they recognize. NIH's basic definition of success rate is the percentage of reviewed grant applications that receive funding. Success rates are determined by dividing the number of competing applications funded by the sum of the total number of competing applications reviewed and then the number of funded carryovers. Funded carryovers are those applications that were reviewed and not funded in the review year, but were funded in the next year. In the review year, the application is counted only in the success rate denominator, but in the next year when the application is actually funded, it's included in the success rate numerator and denominator. And so applications having one or more submissions for the same project in the same fiscal year are only counted once. Some grants are jointly funded by two or more NIH institutes. Typically, the institute that contributes the most dollars to the grant receives the award count, but the dollars contributed by each IC are reported. Excluded from the calculation of success rates are those applications that are withdrawn by an applicant prior to review, or returned or administratively withdrawn by the NIH Center for Scientific Review, or an NIH institute and not peer reviewed by an initial review group. And as you can see from the slide, NEI's 2019 success rate has increased to 28%, while NIH's success rate has remained constant at 20%.
Slide, NEI operating levels versus buying power, FY 1999 through FY 2019. Bar chart shows doubling period of NIH budget from 1999 to 2003, where buying power is at its highest level. Operating budget consistently increases from 2014 to 2019, while buying power remains relatively flat.
Here you have the NEI operating levels versus buying power from 1999 through 2019. This multicolored bar chart shows a comparison of current day buying power to the late 1990s and early 2000s, when we experienced the doubling of the NIH budget. The NEI operating budget is represented by the green bars on the left. The buying power relative to the operating levels is represented by the blue bars to the right. And as a reminder, our operating level is our appropriation minus any rescissions or transfers that reduce the amount that Congress has appropriated. So it's basically the funds that we have in hand to operate with. A rescission is the cancellation of budget authority previously provided by Congress. Funds can be taken from us by the Office of Management and Budget, which is the President's budget office, in the form of an across-the-board reduction. And then there are also other transfers that may be unique to the Department of Health and Human Services and NIH. The take-home message from this chart is that although we continue to see modest funding increases over time, inflation continues to significantly impact the value of our dollars, specific to the goods and services procured to support NIH research.
Slide, 21st Century Cures Act. Table, NIH innovation projects in millions, listed from FY 2017 through FY 2026. The last number is not to exceed amount. Precision Medicine Initiative, 40, 100, 186, 149, 109, 150, 419, 235, 36, 31, and 1,455. BRAIN, 10, 86, 115, 140, 100, 152, 450, 172, 91, 195, and 1,511. Cancer Moonshot, 300, 300, 400, 195, 195, 194, 216, NA, 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 and 1,800. Regenerative Medicine, 2, 10, 10, 8, 0, 0, 0, 0, 0, 0, and 30. Total, 352, 496, 711, 492, 404, 496, 1,085, 407, 127, 226, and 4,796. FY 2020 is highlighted. Note, funds not available until appropriated by Congress each fiscal year. Amounts appropriated cannot exceed what is authorized for each fiscal year, and funds are available until expended.
And finally, this is a reminder of the 21st Century Cures Act. The Cures Act and NIH Innovation Fund projects are projected to be appropriated $5 billion over 10 fiscal years. The NIH Innovation Fund projects are Precision Medicine, which is now called the All of Us Research Program, BRAIN, Cancer Moonshot, funded by the National Cancer Institute, and Regenerative Medicine. And FY 2020 is the final year of the Cures Regenerative Medicine project.
Slide, budget process timeline during a normal budget year.
So before I get into the funding for 2020, I imagine that there would be some participating on the call today who are not completely familiar with how the federal budget process plays out. So I've provided here a budget process timeline, but keep in mind, this is a timeline during a normal budget year, which is actually very rare these days. The budget process is complex and can be thrown off track by events that are happening in the nation, like what we're experiencing now with the coronavirus pandemic, or whether or not it's an election year. This year, we have both. So predicting what Congress might do by way of when and how much our budget might be is not easy. We do try to read the tea leaves, relying heavily on information that comes out of these well-defined actions that play out during the budget process. And so starting with the early fall, agencies like the Department of Health and Human Services, of which NIH is an operating division, submit budget requests to the Office of Management and Budget, which again is the President's budget office. OMB is tasked with implementing the President's budget priorities across the executive branch. In November, typically around Thanksgiving, OMB provides a formal response to the federal agencies on their budget submissions. Initial decisions based on the President's priorities are made by OMB and passed back to the federal agencies. In December, agencies incorporate revisions from the pass back and submit final budget requests to OMB. Then next comes the State of the Union address delivered by the President to a joint session of Congress, at which time budget and other priorities are shared. Then we move to the release of the President's budget. By law, the President is required to submit the budget request between the first Monday in January and the first Monday in February. In this era, the first Monday in February is typically when the President's budget is released, unless there is some intervening situation that makes the timing impossible. For FY 2020, for example, the budget release was delayed due to the federal shutdown. Then the spring ushers in the period for Congress to develop and pass the appropriations for the upcoming fiscal year. In April, the Senate introduces legislative measures to deal with issues affecting both houses of Congress and may create temporary joint committees. April is also the deadline for Congress to pass its budget resolution to guide decision-making for the 12 appropriation subcommittees. In May, the House of Representatives starts considering annual appropriation bills, with June 10th as the deadline for the House Appropriations Committee to submit its last annual appropriation bill to committees. The end of June is the deadline for the House to approve annual appropriations bills. The FY 2020 appropriations process has been greatly delayed by the coronavirus pandemic. The House and Senate are expected to start markups in late June. Markup is the process by which congressional committees and subcommittees debate, amend, and rewrite proposed legislation. Then fast forward to October 1st, and the new fiscal year starts whether we have an appropriation or not. And so without an appropriation, we may be faced with a shutdown or be under temporary or full year continuing resolution. But of course, we always hope that we will have a full year appropriation. And a glimmer of hope is that traditionally in an election year, Congress does not like to anger voters with a shutdown.
Slide, FY 2020 President's Budget versus Enacted Appropriation. Table, NIH versus NEI, listed are FY 2019 enacted, FY 2020 President's Budget, percent change, FY 20 PB versus FY 19. FY 2020 House, FY 2020 Senate, FY 2020 Final Bill, percent change FY 20 versus FY 19, and dollar change FY 20 versus FY 19. NIH in billions of dollars, 39.08, 33.9, negative 13%, 41.15, 42.08, 41.68, positive 6.7%. 2.6. NEI in millions, 796.5, 685.6, negative 14%. 835.5, 840.2, 824.1, plus 3.5%. 27.6. FY 2020 final bill is highlighted. FY 2019 enacted is not adjusted for transfers and excludes supplemental funding. FY 2020 final bill does not include reductions for rescissions and transfers. This table shows the history of the NIH and NEI appropriations from last year's budget in 2019 through the final 2020 appropriation. Each step in the process is significant because it gives insight into what both the President and Congress sees as funding priorities. The current administration has consistently requested significant cuts to the NIH and NEI budgets. The House and Senate, however, tend to prioritize NIH research and propose increases because they see the direct impact of NIH-funded research in their communities and across their states. The mission of NIH in general and the research focuses of our institutes and centers is something that members of Congress and their constituents understand. As displayed in the table, had the current administration gotten its way, the NIH budget would have been cut by 13% and the NEI budget would have been cut by 14%. The President's budget is a planning document, and we know that the power of the purse sits with Congress. However, until Congress gives some indication about their posture for the upcoming year, the President's budget request, no matter how deep the cuts, looms large over our planning process. Ultimately, in FY 2020, NIH received a 6.7% or $2.6 billion increase, and NEI received a 3.5% or $27.6 million increase over FY 2019. Slide, NIH appropriations since sequestration. Table in billions shown in the following sequence. Fiscal year, appropriation, dollar change and percent change. 2020, 41.7, 2.6, and 6.7%. 2019, 39.1, 1.8, and 4.8%. 2018, 37.3, 3.0, and 8.7%. 2017, 34.3, 2.0, and 6.2%. 2016, 32.3, 2.0, and 6.6%. 2015, 30.3, 0.2, and 0.7%. 2014, 30.1, 0.8, and 2.7%. 2013, 29.3, NA, and NA. The next two slides are funding tables that show the progression of the NIH and NEI budgets for each year since sequestration in 2013. It took until 2016 for NEI to get back to pre-sequestration levels. Since then, we've seen increases each year. However, with funding increases comes the expectation from Congress that NIH and NEI in turn will increase the number of competing awards. NIH and NEI strive to meet those expectations while allowing for the realities of funding and increasing commitment base. Slide, FY 2020 operating level, estimated spending by funding mechanism. Table, funding mechanism followed by amount in millions. Research project grants, 513.2. Small business, 25.5. Research centers, 27.4. Other research, 76.7. Training grants, 12.2. R&D contracts, 43.8. Subtotal, extramural research, 698.8. Intramural research, 93.5. Research support, 31.1, and total NEI, 823.3. Bar chart showing percent share. This slide contains a funding table and a bar graph. The table to the left shows the projected amounts to be spent this fiscal year in each funding mechanism. The funding mechanisms are research project grants, small business awards, research centers, other research, which includes career awards, conference grants, resource grants, and cooperative agreements. Training grants, research and development contracts, intramural research, and research support. The bar chart to the right shows the same information, but at a higher level corresponding to the major funding mechanisms. The major mechanisms are extramural research, intramural research, and research support.
85% or $698.8 million of NEI's FY 2020 operating budget is distributed to external research activities, including all grant and contract mechanisms. 11% or $93.5 million is estimated to be spent on intramural research. The intramural research program funds internal principal investigators and fellows conducting basic, translational, and clinical research. The remaining funds at 4% or $31.1 million covers administrative, logistical, and scientific support for the NEI programs and includes administrative functions such as awarding grants and contracts, planning and evaluation, and other management activities. For some time, NEI has consistently held to the same % share of the budget for each major funding mechanism, which is in line with the NIH overall.
Slide, FY 2020 supplemental funding bills.
Here we have the FY 2020 supplemental funding bills that have been passed this fiscal year. So as a result of the coronavirus pandemic, within the last few months, Congress has passed several supplemental appropriations bills. The passage of these supplemental bills for response and relief efforts demonstrates how quickly and decisively Congress can move during a state of emergency. The first bill is the Coronavirus Preparedness and Response Supplemental Appropriation Act, which passed with near unanimous support in both the House and the Senate. This bill provides $8.3 billion in emergency funding for federal agencies to respond to the coronavirus outbreak. Of that $8.3 billion, $6.7 billion is designated for domestic response and $1.6 billion for international response. The majority of the funding for the domestic response, which was $6.2 billion, actually went to the Department of Health and Human Services, $3.4 billion to the Public Health and Social Services Emergency Fund, $1.9 billion to the CDC, $836 million to the National Institute of Allergy and Infectious Diseases, which conducts research on therapies, vaccines, diagnostics, and other health technologies here at NIH, and $61 million to the FDA for the development and review of vaccines, therapeutics, medical devices, countermeasures, and to address potential supply chain interruptions. The bill specified that funding could be distributed over a multi-year period, and the period of funds availability varies by the organization receiving the funding. Next, on March 18th, 2020, the Families First Coronavirus Response Act was signed into law, making the second major legislative initiative to address COVID-19. The total funding for this initiative is $3.5 billion, of which $1.3 billion was directed to the Department of Health and Human Services, with $250 million going to the Administration for Community Living, $64 million to the Indian Health Service, and $1 billion to the Office of the Secretary for the Public Health and Social Services Emergency Fund, which supports the National Disaster Medical System to pay claims of providers for detection and diagnosis of COVID-19 and testing related visits for the uninsured. On March 27th, 2020, the Coronavirus Aid, Relief, and Economic Security, or CARES Act, was signed into law, marking the third and largest major legislative initiative to address COVID-19 to date. The cost of the CARES Act is approximately $2 trillion and contains a number of health-related provisions focused on the outbreak in the United States, including paid sick leave, insurance coverage of coronavirus testing, nutrition assistance, and other program efforts. Included in the bill is an additional $945 million for NIH to expand prior research plans, including developing an improved understanding of the prevalence of COVID-19, its transmission, the natural history of infection, and novel approaches to diagnosing the disease and past infection, and developing countermeasures for the prevention and treatment of its various stages. On April 23rd, an interim coronavirus relief bill called the Paycheck Protection Program and Healthcare Enhancement Act was signed into law, marking the fourth major legislative initiative to address COVID-19. The bill included $1.8 billion for NIH to accelerate development of point of care and rapid diagnostic technologies, $306 million of which to be transferred to the National Cancer Institute, $500 million to the National Institute of Biomedical Imaging and Bioengineering, and $1 billion to the NIH Office of the Director, with transfer authority to the accounts of the institutes and centers of the NIH. The HEROES Act, which was signed into law on May 15th, provides an additional $500 million for the National Institute of Allergy and Infectious Diseases, $200 million for the National Institute of Mental Health, and $4.021 billion to the NIH Office of the Director to prevent, prepare for, and respond to coronavirus domestically and internationally. $3 billion of the amount provided is designated for offsetting the costs related to reductions in lab productivity resulting from the coronavirus pandemic or public health measures related to the coronavirus pandemic, and $1.021 billion in transfer authority to the institutes and centers. All funds are to remain available through September 30th, 2024. And finally, there is a spending bill pending legislation for additional coronavirus relief that has passed the House of Representatives, but not the Senate. The Senate is holding on that legislation as they determine how the funds that have already been approved are being managed.
Slide, FY 2021 President's Budget Request. Table, funding mechanism followed by amount in millions. Research project grants, 462.3. Small business, 23.2. Research centers, 24.9. Other research, 69.8. Training grants, 12.2. R&D contracts, 42.8. Subtotal, extramural research, 635.2. Intramural research, 84.9. Research support, 28.9, and total NEI, 749.0.
And so now moving to the outlook for FY 2021. Here we have a table that divides the proposed 2021 President's budget for NEI by funding mechanism. The President, via the Office of Management and Budget, proposed a 7% reduction to the NIH budget and a 9% reduction to NEI. If this budget request were more than a planning document and actually came to fruition, NEI would have to cut approximately 125 awards across all grant activities except for NRSA training slots, and cut approximately $63 million from the extramural research budget. Given that Congress controls the final funding amounts and not the President, drastic cuts like this are not expected in 2021.
Slide, FY 2021 budget process.
This slide is just a snapshot of where we are in the 2021 budget process. As I mentioned previously, the appropriations process has been delayed significantly by the coronavirus pandemic. However, Congress has started to adopt new rules allowing for remote committee markups, given that they're expected to start that process very soon.
Slide, contact information. Karen Colbert, NEI Budget Officer. karen.colbert@nih.gov, 301-435-8822. NEI Financial Management Branch, neifmb@mail.nih.gov, 301-496-5243.
And I have gotten to the end of my slides. This is my contact information. And if at any time you ever have questions about NEI's budget, please feel free to reach out to me at any time.
Thank you, Karen. Are there any questions from panelists for Karen? I will tell you that with the permission of the presenters, I convert their slides into PDF files and I post those on the electronic council book. So you will have access to her slides and the information there. I can also send an email to council members with the link to the NIH's statement on sexual harassment.
Okay, Thank you. I'll stop sharing.
So are there questions for Karen on the budget?
There's going to be appropriation for loss of lab productivity. And I was wondering if there was a plan how that would be implemented.
As you know, this is a very fluid process, and I will say that at this moment, I cannot say, how funds will be distributed, for the intramural program. We do know that the kind of loss of productivity within our labs, it's something that is crucial to NIH and something that we know that NIH leadership is addressing, but how that will play out within the budget process, I actually cannot say at this moment.
Okay, thank you.
Are there any other questions? If not, thank you very much, Karen.
You're very welcome. Thank you.
Okay. And so next we're going to hear from Russ Van Gelder. Russ, as a member of the Council of Councils, maybe you can just give us a very brief update on the past two meetings.
Great. Thank you, Annie. I don't have slides for this, and I will try to keep it fairly brief. So as Santa mentioned at the beginning, the Council of Councils is an arm of the Division of Program Coordination, Planning and Strategic Initiatives, or DPCIPSI, that was established in 2006 in the NIH Reform Act of that year. The basic idea was that the director of the NIH, Francis Collins, currently felt that although the 27 institutes and centers that make up the NIH had independent funding, the central office really had almost no resources to coordinate across the NIH. And in the 2006 Act, among other things, the Common Fund, which is the direct funding to the director's office, as well as the Council of Councils, were specified. The way this council works is each of the 27 institutes and centers that make up the NIH will nominate an individual, or actually nominate at least 3 individuals for a position on the Council of Councils. And so this is essentially a body made up of nominees from the individual institutes. And I was very fortunate to be nominated and selected this past year. The terms on this council are long. It's actually a 6-year term, and the counselors are approved actually by the Secretary of Health and Human Services. So the vetting on this was pretty extensive. I thought the vetting was pretty extensive for any NAEC, but the Council of Councils was a whole nother level up in terms of the vetting process. Jim Anderson, who's Deputy Director of NIH, is the DPCIPSI, which is the NIH speak acronym for this office, is the head of DPCIPSI and chairs the Council of Councils, and Francisca Greider is the executive secretary. The Council of Councils has 2 functions. One is a sort of administrative function in that we serve as the council for the Office of Research Infrastructure and Programs, or ORIP, which is a little bit of a catch-all for a number of programs that don't fit in any single institute. Most predominantly the primate centers, the national primate centers, are administered through ORIP, and we serve the same function for ORIP as our current council does for the NEI. But the larger work of the Council of Councils is really to review common fund initiatives, that is, programs that require multi-institute and center coordination and strategic planning. Currently, the Common Fund has a budget somewhere a little bit north of $600 million a year, so it's slightly smaller than the NEI in terms of a budget, but its programs affect all of the institutes. So my goal just in the last 2 or 3 minutes here is to, number one, give you just a flavor of the types of things that are discussed. I've had the opportunity to attend 2 Council of Council meetings so far, one in person in January. I was not yet vetted through the process, and then we had a remote meeting last month in May for which I was a voting member of the council. And I want to close then by really asking what the best mechanisms are for coordination between this council, NAEC, and Council of Councils to ensure that our voice, that is NEI's voice, is heard at that larger table. So among the items that were discussed at the last two council meetings include a common fund FOA reissue, similar to the reissue that we just approved for our K and R24 awards. The Council of Councils approved a Pioneer Award, a New Innovator Award, Transformative Research Award, and Early Independence Award. These are all large awards administered through the Office of the Director. These are not new awards. This was a reissue, but there was some review of those awards and their impact to date. There was also a reissue of an HIV Scholars Award through ORIP for K01s and R21s. And then much of the work that we do are trans-NIH initiatives. For example, a discussion of a working group on DNA sequence reads and the best way to annotate and ensure that we have a working repository of all the sequence coming out of various deep sequencing initiatives. There was a common fund concept clearance in May on the design and use of artificial intelligence platforms for biomedical and behavioral research, including some very critical decisions in terms of common platform description for large data sets and whether or not NIH would actually host data sets. That decision was not to host data sets, but to provide a common protocol for submitting data sets so that there's interoperability. We had a clearance on an Office of Dietary Supplements concept for administrative research supplements for research on dietary supplements. Obviously, some of the research within the NEI on AREDS type vitamins falls under that, that umbrella. We had several working groups within the Council of Councils, including a new basic behavioral and social science research working group. I made a plea that sensory deprivation diseases such as blindness and deafness be considered within behavioral research for their impact in that area. We also spend a fair amount of time talking about issues that are trans-NIH, including sexual and gender minority research, strategic planning, and concept clearances on programs to improve equity, diversity, and inclusion within the NIH. There was a significant discussion led by the NIH Chief Officer for Scientific Workforce Diversity, Dr. Valentine, on a concept clearance for faculty institutional recruitment for sustainable transformation. It was a really interesting discussion. Essentially, this is a program to set up centers of excellence for the advancement of scientists from underrepresented minority backgrounds and essentially create critical masses at specific institutions large enough to support a vibrant community. A little bit controversial in that the money that would go into this is substantial, on the order of $250 million. And that would include recruiting money for those select institutions. And there's some concern that that may result in actually decrease in diversity at other institutions if there is cross-institutional recruitment. So that's just a flavor for the type of questions that we have been addressing at the Council of Councils. It's pretty heady stuff. These concept clearances generally take about 15 minutes and often involve hundreds of millions of dollars of potential programmatic budget. And my concern in representing the NEI in this capacity is that this council, the National Advisory Eye Council, has a voice at this larger table. As Karen just showed, our budget is perhaps 2% of the NIH budget. And so we're not big players at the table, and there's representatives from NCI and some of the other very large institutes. There are 27 voting council members, so it's about twice the size of our own council. And it's challenging to really get our voice heard even at this larger council. And so I'd like to coordinate to the extent I can with our council to ensure that I am speaking up for those initiatives that we are most invested in. As such, I would propose that I'd be happy to give a short executive summary of the Council of Councils activities. Obviously, as with our own council, the whole recordings are transcribed. There are minutes and the whole thing is recorded, but it is typically a 6 or 7 hour meeting, and I don't think anyone has time to watch the whole proceedings unless they have a strong interest in this council. So I would like to, as sort of a consent agenda type practice, give an update on the activities that came before Council of Councils, open up an open channel of dialogue between any council member who wants to reach out to me, particularly after I rotate off. The I-Advisory Council next January is scheduled, and if history is a guide, probably next May. And I also want to ensure that I have an open dialogue with the Director of Extramural Activities and the Director or Acting Director of the NEI, again, to ensure that our voice is heard at this larger table. So I'll stop there and happy to take any questions on that.
So are there any questions for Russ? We can keep some discussion of this, I think, for the general council discussion, so we could just move ahead, but if there's any pressing questions. But Russ, one thing I will say is that we can definitely get you back as a speaker at our future council meetings so that you can continue to be that active liaison for us.
Thank you, Annie. I would appreciate that. I think we've had historically a bit of a disconnect between the Council of Councils and the National Advisory Eye Council, in that there has not been an open and established channel for that communication. And I would really like to have that channel open, because a lot of things do happen at this Council of Councils level that impact the NEI. And I would very much appreciate the advice of this council that I've been privileged to serve on the last 4 years as I represent our views to the larger NIH community.
Okay, thank you very much. If there's no pressing questions, we can definitely continue this conversation in the general council discussion. But right now, we've got one more talk before we take a break. And that's going to be Shefa Gordon. Dr. Gordon is the director of the Office of Program Planning and Analysis, and he has an update for us on program planning activities.
Slide 2020 Vision for the Future, NEI Strategic Planning Update.
Go ahead.
Good morning, everyone. My name is Shefa Gordon, and I'm the director of the NEI's Office of Program Planning and Analysis. I have a lot to cover today, but I know we're running a little bit late, and I'm the only thing between you and a break. So I guess the benefit of a virtual council is that you can get up and stretch your legs or get a glass of water if you want. But I'll also try to keep this quick. We are past halfway through developing our next NEI strategic plan, which we're calling 2020 Vision for the Future. We're starting with a broad information gathering phase, as Dr. Tumminia mentioned, which once we get a new NEI director, we'll be developing our plan in earnest and starting with your implementation phase of our planning process. At Council in January, I presented on the first step of planning, or a public request for information, RFI, which generated over 250 responses. We were very pleased with that response, and we used that input in assembling expert panels for our each of our areas of emphasis. Today, I want to focus on those panels. I haven't talked about who they are and what they've accomplished.
Slide, Areas of Emphasis.
So our areas of emphasis, we have 7 areas of emphasis arranged in 3 research domains. The first domain is visual system in health and disease.
With areas, from genes to disease mechanisms, biology and neuroscience of vision, and immune system and eye health.
The second is capitalizing on emerging fields.
With areas, regenerative medicine and data science.
And the third is a public health domain, which is preventing vision loss and enhancing well-being.
With areas, individual quality of life and public health and disparities research. Slide, from genes to disease mechanisms. Shown first are portraits of NEI council member who is highlighted, followed by the 2 co-chairs, then followed by an additional 10 panel members.
So as you may remember, the plan has always been to for each panel to meet 3 times for about 90-minute calls over Zoom. And this is before Zoom became trendy. So our planning office was a little planning ahead of schedule. I want to show you what these Zoom calls look like. We asked for, this plan is a product of council. So last year we asked for volunteers for each panel. For the panel called From Genes to Disease Mechanisms, Lou Pasquale volunteered. We then, so he's our council representative. We then selected 2 co-chairs, Vasilis Vasilou and Renu Kaluru, who helped us identify the rest of the panel. Each panel has 12 to 13 experts plus 2 to 5 NEI program or planning staff. I don't have time today to list all 113 individuals who worked on these panels, but we can make these slides available after today's meeting so you can see their names and their faces here. I do want to emphasize that these panels are really all-star panels. I've sat in all but about 2 of the calls so far, and they've been really productive and energizing. So we've been very pleased with our plan, with the panels. We worked really hard to achieve diversity on the panels with regards to gender, race and ethnicity, age, geography, area of expertise, degree. We have people with PhDs, MPHs, optometry, ophthalmology degrees. We have researchers, practicing clinicians, and other members of the public. We have some panelists who are blind, partially vision impaired, or fully sighted. For each panel, we tried to make sure that we covered our major programs. So we have retina, cornea, glaucoma, myopia, all those are represented. And for this panel, from the genes to disease mechanisms, we had experts in rare diseases and gene therapy, including some former council members. So as it turns out, Lou Pasquale actually just rotated off of council last month. But fortunately, Renu Kaluru just rotated on, so we still actually have council represented on this panel.
Slide, From Genes to Disease Mechanisms Preliminary Panel Highlights.
I want to briefly discuss the preliminary panel highlights. I say preliminary because even though this panel has completed all 3 meetings, we're still pouring over 30 to 40 pages of notes and materials we've got from the panels. And we're going to work with the panels and the chairs to distill their recommendations to about 4 pages that will for our final plan. I do find it interesting how we've had some convergence in the recommendations and the things that were discussed. So this plan will discuss databases and how standardized data collection is needed for harmonizing between different databases. And that came up on a lot of different panels and of course was discussed in our data science panel. Another common theme was new model systems.
Centralized support for development of patient-derived iPSCs, organoids, Cree mouse lines, zebrafish lines.
Animal models, and this really is a theme, but we tried to get people specific rather than being generic about we need more animal models. Be specific, what do you need for this type of research? The concept of aging came up in a few different panels. There's a lot of age-related eye diseases. Angiogenesis, we talked about how blood vessel growth during tumor development relates to retinal vascular diseases. Sex hormones.
Explore their role in eye disease.
Redox biology.
Understand mechanisms in eye development and disease.
And we talked about the collaborations in the talent pool to expand interactions beyond ophthalmology and vision research departments.
Slide, Biology and Neuroscience of Vision. Portraits of 13 panel members with NEI council member highlighted.
Moving on to the Biology and Neuroscience of Vision panel. This is the topic that actually received the most comments from our RFI, and we specifically included experts on the topics on cortical visual impairment and visual decisions and behavior, which are topics that came up a lot in the RFI. This panel has met twice, and the third call is scheduled for June 29th. Bill Guido and Monica Vetter are the co-chairs. Jose Manuel Alonso is our council representative. Slide, Biology and Neuroscience of Vision Preliminary Panel Highlights. Just some panel highlights that we recognize that there's the whole brain initiative at NIH, which is developing a lot of new imaging technologies. Every year we've been telling you about the progress of how about 20% of new brain projects actually are related to vision, either retinal or the brain. And we were thinking about, the panel mentioned that we could capitalize on some of the new technologies and actually apply them better to the visual system. They also discussed visual circuit dissection using new tools such as single-cell RNA-seq to catalyze cell subtypes, gene programs, to couple that with multicellular recording and to do 3D reconstruction of synapses, to use some new tools for looking at the molecular responses, to explore plasticity, to develop tools to modulate plasticity in development and repair. But then cortical visual impairment, CVI, was, again, this topic that I brought up as a big issue that came at the, in the RFI. These are often strokes in utero, but there's it's a kind of a broad term, and we do not really have functional assays to define and diagnose this broad family. So the neuro panel decided that it would be recommended that we should really get a better handle on what we're looking at. And also looking at the visual decision making to build on visual processing research to really model higher levels of brain function.
Slide, Immune System and Eye Health. Portraits of 13 panel members with NEI Council member highlighted.
The Immune System and Eye Health panel was charged with looking at immunology, inflammation, and infection across the visual system beyond the ocular surface, which comprises the bulk of our current portfolio. It was chaired by David Lieb and Jennifer Thorne, with, of course, strong input from our council member, Russ Van Gelder. We've had 2 meetings, and the final call is scheduled for July 6th.
Slide, Immune System and Eye Health preliminary panel highlights.
We were again building off, our first call built off things in the RFI, looking at the microbiome. We looked at the gut microbiome and its role in regulating homeostasis, and then even thought to really use artificial intelligence for personalized medicine to kind of figure out what species in the microbiome are having what interactions with the immune system. We talked about infectious eye diseases, herpes, uveitis, Ebola, and even SARS-CoV were discussed. Looking at AMD and chronic degenerative diseases. How do chronic diseases differ from inflammatory diseases? Talking about immunotherapy. Consider how the acute versus chronic stimuli and stimulating versus regulatory responses. An idea to look at alternative therapies to steroids, particularly for uveitis. We discussed immunosenescence and looking at how the immune system, we have a lot of aging eye diseases, and how the immune system is really changing over the course of your lifespan. So what is the impact of both the immune environment and aging on the immune, ocular immunology? A functional mapping of genes, different genes and their role in immune pathways. We also talked about neuroimmunology and the immune privilege. There's a lot of lessons that can be learned from the brain, such as where are the lymphocytes getting into the system. In the brain, there are CD8 positive cells, the meninges. There are particular beneficial impacts of cytokines, such as the effect of IFN gamma actually impacts neuron behavior. Are there similar effects on the retinal, on the visual system like there are in the brain or in the retina, I guess? And we also talked about live quantitative imaging of ocular immune responses.
Slide, Regenerative Medicine. Portraits of 13 panel members with NEI Council members highlighted.
The Regenerative Medicine panel has 2 volunteers from Council, Katia Del Rio Tsonis and Marco Zarbin, and along with our co-chairs, Val Cantosolar and Jeff Goldberg, they've ably led this panel. For this particular panel, we have expertise that includes industry experts, engineers, and researchers who have worked with the FDA to approve new products. We were building off existing NEI regenerative medicine programs like the Audacious Goal Initiative and 3D Retina Organoid Challenge, but what are the next challenges for NEI? And naturally, our new NEI Office of Regenerative Medicine with Amber Reed and Steve Becker took the lead on organizing this particular panel.
Slide, Regenerative Medicine preliminary panel highlights.
Some of the highlights from this panel is really looking at the human immune response to understand how the response to cell and tissue transplants under different disease conditions and surgical methods. Material transfer can be a problem for certain types of understanding the effects of retinal transplantation, but the goal is to understand and harness material transfer in photoreceptor precursors and extracellular vesicles from other cell types for therapeutic benefit. Extracellular vesicles, that concept is something that also came up in the RFI. We made sure to include some expertise of that on our panel. Looking at automation and artificial intelligence to apply new technology to improve cell manufacturing capacity and quality. Directory programming, supporting techniques to differentiate cells and other cell types using biological and chemical factors, such as converting Mueller cells into rods, RPE into retinal neurons, or rods into cones. And also looking at retinal organoids, extracellular vesicles, and biomaterials. There's a lot of advances in that area.
Slide, data science. Portraits of 13 panel members with NEI council member highlighted.
The data science panel was a bit unique in that we didn't really have much of an existing program area in that yet. And it was a little bit harder to form this panel. They had their first meeting a few weeks ago, and they're scheduled for their second meeting next week, and a third meeting will take place in July. You may remember last January council, there was a fantastic production by Aaron Lee. He's one of the co-chair for this panel. Sebastian Sung, who's also on the panel, also gave a council presentation a few years ago. The other co-chair is Michael Chang, who led the development on a tool that was FDA approved this January that enables diagnosis of retinopathy prematurity using an AI algorithm for telemedicine. Dennis Levy is the council representative. This panel has a mix of expertise as well, including academic data scientists, data generators, and industry, and a bioethicist from the Patient-Centered Outcomes Research Institute, PCORI, to look at the bioethics concerns in data science.
Slide, data science preliminary panel highlights.
So there is NIH just recently put out a NIH-wide data science plan. So we didn't feel the need to reinvent the wheel, but we did want to look how NEI could build off that and as we develop our own data science strategies. The big topics that came up were big data. Vision can actually lead the way here. There are a lot of unique data types, and we're positioned. We already are kind of a leader in data science, particularly in the AI domain. But any access to clinical imaging and genetic databases must address privacy concerns, such as protected health information and personally identifiable information. We need to come up with data sharing incentives and address the lack of standardization. If you're de-identifying image data, how do you do that? How do you really de-identify image data so that it can't really be traced back to the patient? There's storage and format standardization issues as well. We talked about artificial intelligence.
Barriers include standardizing data and combining disparate data types.
Computational advances, so investing in good code and making sure we annotate that code so we don't lose the good data collection.
Review process for methodology to avoid wasting good data collection. Common data repository for large data sets.
And training in workforce is common, of course, a lot of areas in vision science, but particularly in data science, we're competing with Googles and Facebooks of the world. So we really need to find a good way to recruit and train new talent and incentivize them to work in our field. One thing, when Aaron Lee presented in January, council actually came up with this idea, the centralized cores or centers that data scientists can work with data generators to really create a center for training and rather than having data generators that may not be able to develop the artificial intelligence themselves, to really work together in close collaboration with some of the data scientists. So that was the last idea, something that was actually came from council and we suggested to the committee, and they endorsed it.
Slide, individual quality of life. Portraits for 13 panel members with NEI council members highlighted.
Another council presentation you remember from, if you were around 3 or 4 years ago, was Lotfi Merabit, who works on CVI. He's one of the co-chairs for the individual quality of life panel, along with Ava Bittner, and Emmy Hartnett is our council lead. Although we now welcome James Coughlin to council, who is also on this panel. So we have 2 representatives from council on this panel. This panel has really diverse expertise, from rehabilitation research to expertise in mental health and social isolation, to assistive device developers, to epidemiology, to low vision educators, schools for the blind. We have Veterans Affairs and American Federation for the Blind representation on this panel.
Slide, individual quality of life preliminary panel highlights.
They really were very busy, and I'm just going to run through some of the things they talked about. So brain-related visual impairment, so CVI, kind of the rehabilitation and diagnosis end of things, as well as traumatic brain injury, stroke, and neuroscience, plasticity, telemedicine, better understanding of comorbidities and management, really dealing with mental health and wellness. So screening, understanding, and treating for mental health issues, particularly for newly diagnosed people losing their vision, and strategies to promote well-being, technology and communications,
improving efficient access to mobile-based applications, websites, and graphics,
rehabilitation,
develop evidence-based practices,
navigator, if someone is again new, to have an individual who can really be an assistant to help people with low vision navigate the resources that are available to them. Talking about education, identifying predictors of academic success, and establish importance of various educational tools. Discussed employment, establishing reliable statistics, identify predictors of success and barriers to employment, and driving navigation as promoting independence.
Slide, public health and disparities research. Portraits of 12 panel members with NEI council member highlighted.
Finally, our public health and disparities research panel focused on epidemiology, health services, and health disparities research. In addition to epidemiology, community health, and clinical backgrounds, we had health economists from the Veterans Affairs Department, and a biostatistician from the Indian Health Service. For both this and the public health domain, and the individual quality of life panel, we had panelists who had helped develop the 2016 NASM report on population health. So that served as a guidepost for this panel.
Slide, public health and disparities research preliminary panel highlights.
Some of the things that this panel discussed was updating epidemiological data, particularly pediatric and underserved populations, behavioral economics,
studies to generate evidence for the value of including vision health and population health policies,
cost effectiveness research to really demonstrate cost savings and improved health outcomes for interventions such as low vision rehabilitation. Is there, what's the cost savings there? Provisions including eye care to vulnerable populations, diagnostics such as screening, technology, and policy. Developing tools to increase access to care. Telemedicine is one tool, but that's not good for everyone because not everyone has access to the computers or smartphones to really make that work. Harmonized study designs and outcome measures to rigorous studies to really improve systematic reviews, policies, and practices. Health behavior research and care delivery model to really understand patient adherence, what causes someone to follow up on needed treatment, utilize available treatments or health services. Diversity of NEI supported researchers are really helpful for conducting epidemiological, qualitative, and minority focused research, and really community engagement, social determinants of health, and health literacy.
Strategies to better address underserved and minority populations. Slide, timeline and next steps. Gantt chart from July 2019 through January 2021. Published period from December 2020 through January 2021 is highlighted.
So I just want to end by giving you a quick timeline of where we are, next steps. So I'm showing a Gantt chart here with our steps and some dates. We're right here. Here's today's council meeting.
Pointer on Gantt chart shows June 2020 council meeting.
And we just have 4 more of our panel meetings over the next 2 weeks or so. We've already started putting panel reports, writing some of the narrative, and then we're going to start an internal review process, or panels going to review, and then an internal NEI review process, and hopefully to begin layout. We'd like to provide, the goal is to provide a draft of our plan by the next council meeting in October, give you a week or so to review that before council. A lot of this depends on input from our new incoming director when that person gets announced, and any input if they decide that we need to add a new panel, have a new direction. We're certainly happy to figure out how we can improve our plan, and then hopefully we can publish. Since it is the 2020 Vision for the Future, we'd like to publish somewhere around the end of 2020.
Slide, council comments.
So with that, are there any council comments or questions?
Any questions? Thank you, Shefa. That was a tour de force. A lot of information in that talk, and we worked Jen pretty hard the last hour. So, are there any questions for Shefa? No questions. Okay, we're going to look forward to that publication, and hopefully that will be ready before our next council. Thank you so much, Shefa. So at this point, we're ready to take about a 15-minute break. So let's try and reconvene a little bit before 12. And Mike, you can probably stop recording, or you can pause the recording at this point. And let's reconvene at about 11:55, okay?
Okay, recording is resumed.
Okay, so I'm assuming that we've got everybody back on board, and we're going to hear first during this session from Dr. Steve Becker. He's the director of the Office of Regenerative Medicine at the NEI, and Steve has an update on current AGI and RMIP, Regenerative Medicine Initiative activities. So Steve, why don't you take it over and share your screen?
Slide, regenerative medicine updates.
Thank you. I'm Stephen Becker, and I'm happy to present updates about our regenerative medicine programs.
Slide, NEI Audacious Goals Initiative, AGI, for regenerative medicine. NEI AGI Audacious Goals Initiative for Regenerative Medicine Envision Identity Mark.
First, for our new council members, I'll remind you that the NEI Audacious Goals Initiative began in 2013 in an effort to catalyze vision research over the next 10 to 15 years. After a development meeting of over 200 folks from vision research community, it was decided that the goal would be to regenerate retinal neurons and their connections that are lost in disease or damaged. NEI works towards this goal by convening meetings and through strategic funding of research.
AGI for Regenerative Medicine Envision timeline from 2013 through 2021. Several meetings and workshops are listed on the bottom of the timeline, with 3 consortia listed above. Functional Imaging Consortium, May 2015, Discovery Consortium, September 2016, and Translational Models Consortium in September 2018.
In this slide, we show the 3 consortia that have been funded. They include our functional imaging projects, our Discovery Consortium, and our translational enabling models. The meetings that we've held over the past 7 years are also depicted, and the latest one was held in February of this year.
Slide, AGI activities.
That workshop was on understanding human retina biology and perception.
February 7, 2020. Chairs, Maureen Knights, University of Washington, and Adriana DePolo, University of Montreal.
It was led by Maureen Knights and Adriana DePolo. I'll briefly summarize some of the takeaways of that workshop in the next slide, but I wanted to note that the pandemic unfortunately delayed our scheduled PI meetings with the Imaging Consortium and the Translation Enabling Consortium. Also, because ARVO was canceled, the planned town hall and panel discussion on the immunology of retinal cells and tissue transplants did not take place. We did, however, meet virtually with our AGI steering committee,
May 4th, 2020.
which includes Russ Van Gelder, Mark Blumenkrantz, Josh Sains, Rachel Wong, and Clive Svensson.
Slide, AGI workshop, understanding human retina biology and perception. 1. Insufficient understanding of the disease process in humans. Understand nature of visual system in healthy, aged, and disease conditions. 2. Lack of understanding of the microenvironment of the degenerating retina. Access to high-quality human tissue is a barrier. Molecular atlas is needed. 3. Phenomenon of material transfer needs to be understood. Elucidate mechanism and standardize criteria to evaluate cell therapies. 4. Work to understand retinal biology and perception must focus on NHPs or humans. Create and characterize diseased patches of retina in non-human primates. 5. Continue effort to maintain and/or enhance survival and function of remaining cells depending on disease modality, e.g. PRCs, RGCs, RPE. Study mechanisms driving cell dysfunction and disease. Multi-pronged approach to increase cell survival, modulate the environment while stimulating cell integration or regeneration to restore visual function.
For those not familiar with our workshop process, we typically try to summarize the workshop's discussion and report, so our co-chairs have provided a brief overview of the main points that they're working on over the summer with other participants as they compile their published report. I won't go into all the details this time, except to say that we had a fruitful discussion between retinal biologists and those in the psychophysics field, and that there's still much to be understood about the disease processes and the microenvironment of the retina in humans and non-human primates.
Slide, AGI Workshop, Immunological Considerations for Retinal Transplants. What is the immune response? How to suppress/modulate the response. Contribution of microglia and astrocytes in retinal degenerative diseases. Human and NHP ocular immune response/immune privilege of the eye during disease. Current human immunosuppression regimens/future immunomodulatory approaches. Lessons learned from cornea transplantation, RPE clinical studies, dopaminergic neuronal transplantation studies, spinal cord injury studies.
I wanted to mention that the next workshop we are pursuing will be on the topic of immunology of retinal transplants, and it will be a virtual meeting. We will be focusing on the nature of the immune responses to the transplant and how the response can either be suppressed or modulated. We will learn from those who have expertise and experience in the transplantation of other ocular tissues and other neuronal cell types.
Slide, NEI Regenerative Medicine Programs.
Next, I wanted to provide some updates on two other programs. The 3D Retinal Organoid Challenge is a prize competition to stimulate improved retinal models that can be used for disease modeling or drug development. The final submission for this prize competition is October 1st, and we'll be holding an informational webinar on the submission process on June 30th from 2 to 3.
The AMD Integrative Biology Initiative. The New York Stem Cell Foundation, NYSCF, has generated the first 11 iPS cell lines containing HTRA1/ARMS2 and CFH mutations, and they are now available. An additional 30 patient-derived iPSC lines from AREDS2 patients will be available by the end of the summer. Clinical information, image data, and genetic information from the AREDS2 cohort will be available soon.
I also wanted to mention the AMD Integrative Biology Initiative that I briefly introduced last council. We now have available 11 induced pluripotent stem cells lines that were generated from AMD patients from the Age-Related Eye Disease Study 2, or AREDS2. Additional cell lines will become available by the end of the summer at the New York Stem Cell Foundation.
Slide, AMD Integrative Biology Initiative. Logo for NYSCF, the New York Stem Cell Foundation Research Institute. Table of NYSCF and NEI ARD IDs.
To dig a little deeper into this resource, these lines will be available to both academic and commercial entities. Isogenic controls in which the risk alleles have been corrected are also being generated and will be available later this year. The full details about these lines can be found on the New York Stem Cell Foundation website and can be ordered by emailing them at repository@nicef.org. Although these lines are available, NEI is exploring how to leverage them.
Slide, AMD Integrative Biology Initiative FOA concept.
And so I wanted to bring to your attention an FOA concept and provide you with a little bit of more background. As you may remember, the Sorry. The AMD Pathobiology Working Group had been formed a few years ago to evaluate the state of the science in AMD research. One of the needs identified for AMD research was improved cell models. We think this new resource at the New York Stem Cell Foundation will help enable the study of disease mechanisms and progression of AMD. Another feature of these cell lines that I should mention that distinguishes it from other repositories is that there are clinical, genetic, and imaging information from the patients that the cells were derived from, and will be available to researchers on a new platform called BRICS. It is hoped that this will allow correlation of observed in vitro cellular phenotypes with genetic and other clinical data from these patients. So I've listed a few of the objectives and will save discussion until the end of my presentation.
Slide, communicating advances and opportunities.
Also, last council, I mentioned the launch of the NEI Office of Regenerative Medicine. This office has organized a new virtual seminar series called the Vision Innovation Seminars, which will highlight recent published advances by trainees and junior investigators. We will also be starting distribution of e-newsletters via our listservs that highlight relevant publications, funding opportunities, and related programs of interest. We are working on adding content to the office website, and they will have resources and links to data portals, trainees, and for the public to understand more about stem cells. We will also be circulating materials about all these programs at the upcoming International Society for Stem Cell Research annual meeting.
Slide, NIH Regenerative Medicine Innovation Project, RMIP.
Lastly, I wanted to give some updates about the NIH Regenerative Medicine Program, which was funded by the 21st Century Cures Act through 2020. This program aims to accelerate the regenerative medicine field by supporting clinical research. This program has a unique feature in that it requires a match of any federal funding. It also requires participation in an in-depth cell characterization program called the Catalyst. This program characterizes the source stem cells and the final derived products at independent facilities through a contract.
Slide, RMIP Clinical Trial Funding Opportunity Announcement.
This program recently released a funding opportunity for clinical trials. In order to be eligible, the applicant must obtain FDA approval for either an investigational new drug, an IND, or an investigational new device prior to the time of award. The due date for applications is October 2nd.
Connect with us. Sign up for our listservs. NEI, https://go.usa.gov/XPU45. AGI: https://go.usa.gov/XPU4C. 3D space ROC: http://go.usa.gov/XKT3Q. Follow us on Twitter at NATEYEINSTITUTE. Email us neiorm@nei.nih.gov. Portrait, Stephen Becker, PhD, Associate Director, NEI Office of Regenerative Medicine. Portrait, Amber Reed, Program Specialist, NEI Office of Regenerative Medicine.
That's all I have, and I'm happy to answer questions and take comments.
Are there any questions for Steve? Interestingly, in a previous submission for the RMIP, there were two clinical trials that we'll be talking about in our closed session. So that initiative has done good things for these. Steve also is interested in perhaps going forward with the development of one or maybe several FOAs. So right now, there's nothing very specific. There's not been a discussion of set-asides, but I would like to get council approval for Steve and his group to at least move forward with developing those FOAs. And then when there's something more specific, we will bring them back to council. So if I could just get council members, if they approve of Steve moving forward with the development of FOAs in those areas. Steve, you might want to put that slide up again. That was slide 9.
Slide, AMD Integrative Biology Initiative FOA concept.
So in those areas of characterization of cell types, developing protocols, and the correlation studies. So if council members would use the raise hand, and if I could just see whether there's approval for him to move forward with those. And Steve, I'm seeing hands. It looks like there's support for that. Is there any council member who disapproves of Steve moving forward? Let me just lower everyone's hand. So any disapprovals, if you could put your hand up. And seeing none, Steve, then hopefully you can put something a little bit more specific together, and whether it's before or at the October council, we can bring it before council again to get approval on a final form. Okay?
Thanks, Annie.
All right. So at this time, we're going to be presenting 2 concept clearances for RFAs. And first, you're going to hear from Dr. Tom Greenwell. Tom is a program officer who oversees 2 portfolios. One is in the area of central visual development and regeneration, and the second is retinal neuroscience. Tom is also a member of the AGI working group. So Tom, if you would share your screen and talk to the group about the reissue of that U24.
I had to unmute myself. Let me get this slide deck up.
Slide, U24 reissue concept clearance.
Thanks, everybody. Hopefully I won't take too much of your time. As Dr. Schaffner said, I'm here for getting a concept clearance for a reissue of an Audacious Goals Initiative, U24. I am one of the 5 or 6 members of the working group that's in Division of Extramural Science Programs.
Slide, previous NEI AGI FOAs, FY18, content continues across 2 slides.
So the background is that we had an FOA for FY18, and the workshops had underscored an importance of improved model systems and understanding fundamental biology of regenerative medicine processes. A portfolio analysis also supported a need for improved animal models that could be used to assess regenerative strategies. And it would also bring together expertise of groups with expertise in developmental biology, regenerative medicine, retinal degeneration, and other areas. So really a team development and model development. So we put a funding opportunity announcement out for the U24 Translation Enabling Award, and that was released on December 2017. There were five projects funded.
September 2018.
Across the country, containing multiple PI teams, and the models included tree shrew, ground squirrel, and non-human primates. The applicants and the reviewers still communicated.
Tom, this is Anne. Try to talk directly into your microphone. Your voice is fading in and out a little bit for the panelists. So if you could get closer to your mic, maybe that.
Okay. I'll try to do that. Is that better? I can also put my headphones on.
Yeah, it's better.
Okay, great. So, and you can also see my face better. So, yeah, applicants and reviewers communicated that some projects still required more development and preliminary data. So, based on that information, we released an FOA for an R21 award in FY19 for model development. And that was released in December 2018. Remember, the fiscal year starts in October, as Ms. Colbert said. And these awards would help the community generate models and generate more preliminary data for a possible U24 reissue. So for this we got a good response, and we were able to fund 9 projects because they were relatively low cost. And there were a lot more non-human primate models too in this cohort. So we were able to fund tree shrew and novel disease models in mice and non-human primates. And the projects end summer 2020. As you know, COVID has probably delayed some of those projects from getting more data until hopefully the fall or winter, and they will go into no cost extension at the end of the summer. But those projects are moving along, and I've been in communication with those PIs. So the next concept would be to generate models that more closely recapitulate human disease and would be amenable to stem cell approaches. This would create models that also resemble human anatomy and physiology of the retina. And there would be an increased focus this time on non-human primate models because, again, really that's the standard. And as you know, the cells in the retina, in primate retina, both human and non-human primate, are much more similar than cells in a mouse retina. So we want the PIs to test models with appropriate functional assays for integration and also for assessing visual function and improvement. So the proposed funding would be $4 to $5 million for direct costs for 3 to 5 grants.
Slide. Next NEI AGI FOA concept.
Depending on the competitiveness of these projects. So that's all I've really got. If there's any questions, I'm happy to answer them. And I think we're going to call for a vote as well on the concept clearance.
Are there any questions for Tom? I'll wait and see if I see any blue hands go up. So then we would be voting on, council would be voting to approve the reissue of this U24 mechanism for additional, preferably non-human primate models of retinal disease and transplantation. So if I could get council members who approve of the reissue of this U24 to raise their hand. So it looks like, Tom, there is a consensus that you should go forward with this. And I might just ask if there's any council member who disapproves. You could raise your hand now. And seeing none, Tom, you've got your concept clearance. There you go. Thank you.
Great, thank you. It's still weird to do virtual council.
It is. And next we're going to hear from Dr. Martha Flanders. Martha is the program officer, and a director of the Central Visual Processing Program. And she oversees the strabismus, amblyopia, and visual processing portfolio. She is also the director of the Brain Initiative New Concepts R21 program at the NIH. And she's going to talk to you about a possible reissue of the R21 mechanism. So Martha, why don't you go ahead and start?
Slide, goals of the R21 RFA. Possible reissue of RFA-EY-18-001. Biology and Biophysics R01, diagram with 4 right-pointing arrows. Text in arrows from left to right. R21, R01, U01, and U24. 7 multicolored icons representing categories from the BRAIN Initiative.
All right, good. Okay, great. So I'm Martha Flanders. I'm happy to be with you. As Annie said, I'm a neuroscientist and a program officer, and I'd like to talk to you about the possible reissue of an RFA. It's part of the BRAIN Initiative, and it's called New Concepts and Early Stage Research for Large-Scale Recording and Modulation in the Nervous System. And it supports small projects, and we're looking for really innovative new techniques for neural recording and stimulation in the nervous system, anywhere in the nervous system. And so now what you see from the arrows below is that the BRAIN Initiative team that I'm on envisions this R21, the Eye Institute R21, is sort of a part of the open beginning stage of a leaky or open pipeline. These small discovery grants can, if they're successful, allow PIs to continue on to a different RFA, which hosts an R01 for further development, U01 for now bigger groups getting ready for dissemination, which is yet another RFA, and these are all hosted actually by NINDS as part of this team and part of this group. There's also an R01 sort of at the base of all this technology that's aimed at discovering neural mechanisms as nerve cells and glial cells are responding with some of these new tools for stimulation. This biology and biophysics tries to figure out exactly what's going on in the neural tissue. Now the colorful icons below are from the BRAIN Initiative website, and it's just to remind you that you probably already know that the BRAIN Initiative is big. 10 institutes and centers take part in it, and it's also very diverse, cutting across a lot of different areas of neuroscience and neuroscience technology. But let's just focus on this R21 that I want to talk to you about. So,
slide, scope of the current R21 RFA.
On the next slide, I'll talk more about the R21 that we're hoping to reissue. And this R21, it's slightly bigger than most R21s. It's for 2 years, but $300,000 total direct costs across these 2 years. And this has been ongoing for about 5 years now, and we've seen applications for many different techniques: optical, like microscopes, imaging; electrical, for stimulation and recording; a lot of biosensor, really innovative new biosensor tools, and some of the applications are multimodal, combining ultrasound and other modalities, which is kind of neat. On the left, I've got some sort of statistics from the last few rounds. We've been getting about 20 applications in per round. We do it twice a year. What's nice is that about half of the applicants are early stage investigators or new investigators just starting out with their NIH funding, which is really nice to be bringing in new people. And a lot of these new people are from the physical sciences, from physics, material science, electrical engineering, people with chemistry backgrounds. And for these biosensors, there's a lot of protein engineering and genetics going into some of these new tools. And in many cases, it's an investigator from a physical science background that's teamed up with a neurobiology collaborator.
Slide, examples of funded R21 grants.
And I've got a couple examples on the next slide of teams like this, that these are our examples of funded R21 grants in the last few years. And the example on the top is from one of our I Institute PIs, Hae-Young Lee at Hopkins.
1R21EY031265-01.
She's now teamed up with a physics professor, from Hopkins, and they're putting together this non-invasive, low-intensity focused ultrasound, and they're actually using this in a mouse model to stimulate. They're trying to perfect the tool to stimulate in thalamus, and then the Lee lab is already really good at recording activity in visual cortex of the mice, and so they can look exactly at how the stimulation induces particular types of cellular plasticity in mouse visual cortex. The second example is a retina example. I thought you'd like this, so I gave you a picture of the retina, and it's a study that aims at quantifying and really getting a handle on the development of the dopamine system for some of the dopamine neurons in the retina. And so Marla Feller has teamed up at UC Berkeley with Marika Landry.
1R21EY029405-01.
Who's a professor with a chemistry background. And they're developing the kind of biosensor type probe where they can use that retina prep, and they can actually measure and quantify dopamine levels in the retina. And as I said, they're looking at it during retinal development.
Multicolored image shows retinal neural circuits and includes dopaminergic amacrine in black. All amacrine cells transfer rod signals from rod bipolar cells to the axonal endings of cone bipolar cells. Dopaminergic amacrine cells have synapses connected onto the pericardion of all amacrine cells. Source: Massimo Contini and Elio Raviola, PNAS 2003, 100:3:1358-1363.
So I've of course chosen the eye vision research examples to share with you. Of course, this RFA covers technique development for all of neuroscience. It's all over neuroscience, and it's funded through BRAIN, but it's managed right here at the Eye Institute with our review branch, our program branch, and the grants management staff.
Return to slide. Goals of the R21 RFA.
So I'm going to turn it back to Annie for questions or votes.
So Martha, I had one question for you, and maybe an explanation for those who are listening. Your first slide talked about the possible reissue. Is this because the BRAIN Initiative group is unsure of future funding? Or do they feel that they've exhausted a lot of the ideas already with this R21?
That's a great question, Annie. And what's actually going on now in the BRAIN Initiative is we have a brand new director who's just officially came on board a week ago. So we want to let the new director have some time to be sure that he wants to reissue certain successful RFAs that are ongoing will probably be reissued, but we're just sort of in a state where we're in transition. So we didn't want to say for sure. However, it takes a while to get these things going. So we wanted to go ahead and share this with you now to bring it to council.
Okay, thank you, Martha. Are there any questions for Martha about this R21 BRAIN Initiative RFA? The R21s, as you know, are the smaller grants, less money, usually a shorter period of time. And I don't see any hands. So what I would ask is that if the new BRAIN Initiative director decides to go forward with this R21, would you approve of that, since these are applications that are administered by the NEI. All right, so I see a good show of hands. Thank you. And it looks to me like there's consensus. If there's anyone that is opposed, you could raise your hand. So Martha, there's consensus that this R21 reissue would be approved.
Thank you very much.
To go forward. All right, thank you. Very nice talk. So actually, we're down to the end of our presentations. We have one final update on a relatively new initiative, the Anterior Segment Initiative, or ASI. And before I introduce the speakers, Dr. Michael Steinmetz, the director of the Division of Extramural Science Programs, would like just to give a brief prologue to their presentation. So Mike, why don't you unmute and show your video?
Hi, everyone. Can you hear me okay, Annie?
Yes, good.
Thank you. So I just wanted to remind people, you've already heard this from Santa earlier today, but the Anterior Segment Initiative was started, and we had Humam Raj, who spoke to you at the last council meeting prior to our publishing of the request for information. And Humam has done an outstanding job at assembling a group of program staff and members of the review branch and individuals from the NEI-OD to work together, distilling the information in this RFI and incorporating the ongoing discussions that are happening in the program planning panels, and taking a look across our portfolio. And a number of ideas have come forth in this area, including the issues of ocular pain and dry eye. And as you heard from Shefa, the program planning, especially some interest in the microbiome and how it might relate to ocular diseases. And our plan going forward is really to address as many of these issues as we can. But we've decided to focus on, as the first step, the issue of ocular inflammation, because it received a large input through the RFI. So Dr. Sangeeta Bhargava, who is one of our collaborative clinical group, and Dr. George McKee, who's the director of our cornea program.
Slide, Anterior Segment Initiative, Role of Inflammation.
Have put together a little summary of the information that's come from these various sources, and we're going to present it for you today. But you can expect to hear from us at subsequent council meetings as we branch off into other areas. Thank you, Sangeeta. I'll turn it over to you.
Great. Thank you, Mike. Can everyone hear me?
Sangeeta, can I just introduce you and George first?
Yes, please.
Okay. So our two presenters are going to be Dr. Sangeeta Bhargava and George McKee. Sangeeta is a program officer in our collaborative clinical research group. George is a program officer who oversees portfolios in the area of corneal diseases and ocular immunology, inflammation, and infection. Okay.
Okay. Thank you, Annie, and thank you, Mike. Good afternoon, everyone. And in the next 15 minutes or so, George, my colleague, and I will give you a brief description about inflammation with reference to the anterior segment. And, you know, while George and I are giving this presentation, I do want to reiterate Mike's statement that it is not just the work of two of us, but I want to acknowledge a lot of my program colleagues who did contribute in this process.
Slide, Audacious Goals Initiative, AGI. Diagram of 4 connected circles. At center, NEI AGI, the AGI identity mark image. Top circle reads R21, Preliminary Studies for Translation Enabling Models of the Visual System. Right circle reads U24, Translation Enabling Models to Evaluate Survival and Integration of Regenerated Neurons in the Visual System. Bottom circle reads U01, Discovery-Based Science to Identify Factors Influencing Neural Regeneration in the Visual System. Left circle reads U01, Addressing Technical Needs and Opportunities for Imaging the Visual System.
So by this time, all of you are very much familiar with the Audacious Goal Initiative, which is for the regenerative medicine for the retina. This initiative was an effort that was started by the National Eye Institute to push the boundaries of vision science and restore vision through the regeneration of retina. And over the years, the NEI has had multiple town hall meetings, workshops, and came out with many different kinds of FOAs that facilitated collaborative as well as cross-cutting research. And as you can see in this slide, some of the FOAs that came out from the AGI, which were of different kinds of activity codes, whether it was a cooperative agreement or a research grant. And many of this research may have not been possible by using an investigator-initiated application. As Mike pointed out, that in the last council meeting, my colleague Humam gave an excellent talk on the anterior segment, laying a path moving towards the anterior part of the eye. The AGI, as you know, it was more focused on the retina.
Slide, Timeline, right-arrowed timeline diagram, 2013, 2016, 2018, and 2020. Shown later is another right-arrowed timeline diagram, 2019, 2020, pre-COVID, and 2020, during COVID, images of COVID strain.
So if you look into the timeline, and Steve Becker pointed out this earlier, that the AGI was started in 2013, and it's still ongoing where we are right now. And perhaps for the anterior segment, the years might be more memorable in our memory as being in the pre-COVID times, during the COVID times, and hopefully soon in the post-COVID times. So what happened in 2019 December, that is in the pre-COVID times, the NEI put out an RFI, which is a request for information, asking the community to tell us where research should be focused in the anterior segment. And it was literally 2 days before the January Council met that this RFI closed, so we didn't get an opportunity to share with you our responses from the RFI.
Slide, Request for Information, RFI.
We got an overwhelming good response. We got about 52 responses, and it was from 200 stakeholders. These stakeholders were not only from academic institutions, but non-academic institutions, advocacy groups, nonprofit organizations. So it was a really diverse group of people that actually did put their responses for the RFI that we had put out. Several of the responses actually had multiple signatories. And what we also, when we started looking at the RFIs, more than half
52%
of them actually wanted to support the work in inflammation. So over 52% actually wanted some NEI to support work on ocular inflammation with reference to the anterior segment. And the rest, 48%, was divided into these following groups, which was on generating new models for research,
14%,
on drug treatments in infectious diseases,
11%,
on ocular pain.
10%,
There was some on regenerative medicine,
7%,
and there were about 6% that talked about other things on like technology or building an atlas. So there were only 6% that talked about other things. So it was pretty much the the majority had actually emphasized on the inflammation.
Slide, inflammation from RFI responses.
When we looked into the RFI responses, these 52% that we got on inflammation, we wanted to see what really the investigators wanted and what they were really interested in. And these are some of the topics that came out from the RFI responses. They were interested in basic understanding of the inflammation. What is the neuroimmune, neuroinflammation interaction, its role in diseases? What are the model systems? Do we have enough animal models that can recapitulate inflammatory components for human diseases? Do we need better models? Can we use newer technology to understand inflammation, maybe imaging technologies? There was responses that wanted more work on the immunotherapy, on therapeutic agents, and on delivery approaches. There were some responses that were concerning data bank, where you would have clinical samples from human tissues, and that would be something to look at inflammatory biomarkers. And then there was about crosstalk between different mucosal sites. For example, the gut microbiome, that something came out from the RFI responses and its relation with the eye diseases. During the same time in the pre-COVID and also in the COVID times, as Shefa alluded, we were having our program planning, and one of the program planning committee that both George and I have participated is the Immunology and Eye Health.
Slide, program planning comments.
Dr. Russ Van Gelder is our council member representative on this panel, and I want to thank you for your participation on this panel, as well as your valuable comments. And some of the things that really came out from this program planning were very similar comments that we had seen in the RFI, which were, although progress has been made in the field of ocular inflammation, there's still considerable room for advancement. Can we capitalize on newer technologies to understand inflammation? We need novel treatments for ocular inflammation. Is inflammation a risk factor for eye diseases? What is the host immune response in regulating inflammation, in resolution of inflammation? And what are the mechanisms, if there are any, between the gut microbiome as well as in the inflammation in the eye? So it was not only the RFI, the program planning comments that were happening at the same time.
Slide, inflammation, three-circle process diagram. From left, RFI, portfolio analysis, and program planning.
But we also took a look at our portfolio analysis. And we looked at both our basic science immunology portfolio analysis as well as our clinical trial portfolio analysis. And from our basic portfolio analysis, we have about 65 at this present time, 65 active grants, which are focused on the inflammation on the anterior segment. Majority of these are focused on drug treatments to the eye, to the inflammatory diseases. And only 20 of the 65 active grants that we have right now are related to talking about any of the immune components for a specific disease.
Slide, clinicaltrials.gov. Table in following sequence. Subject, total studies, NIH, recruiting plus completed, NEI, NEICC, and NIH, other IC. Cornea, 1,160, 32, 20, 11, and 1. Eye infection, 268, 39, 10, 2, and 27, NIAID. Trichiasis, 12, 3, 3, 0, and 0. Uveitis, 335, 44, 5, 38, and 1. Trachoma, 35, 4, 3, 0, and 1. Ocular herpes, 12, 7, 4, 0, and 3. Keratitis, 698, 17, 11, 5, and 1.
In terms of the clinical portfolio. And this is a table from clinicaltrials.gov, which gives you the different areas of anterior infection that I have taken into consideration: the cornea, the eye infection, uveitis, trachoma, herpes, keratitis. The second column, you can see the total number of studies that have been done, and this is from the clinicaltrials.gov. The third column shows you how many of the completed studies or ongoing studies from the NIH are shown here. The next column shows you the extramural clinical trials that are ongoing, and the next one is the NEI clinical trials from going on at the clinical center. So this is more of our intramural program. And then, of course, the other institutes at the NIH who perhaps may be doing some eye-related work. So for example, when you look at the eye infection here, you see 27 clinical trials from NIAID looking at eye infection. So I went and looked into these 27 trials, and they were all trials talking about drug treatments to HIV. And so I was one of the components they were looking at, whereas we had about 10 of them. If you look at the uveitis field, which is a big inflammatory disease in the anterior segment, there are 38 trials that have been done by our clinical center and only 5 done by our extramural community. So this just gives you a flavor of our portfolio in terms of clinical as well as the basic science.
Slide, detailed analysis.
So when we took all this together, there were 3 themes that came out very clearly, and we kind of binned them into 3 areas, and one was the normal homeostasis, what happens in the disease state of the inflammation, and lastly, how does the resolution of inflammation take place. The one thing that I do want to point out is whether it was in the RFI responses or a program planning, there was a lot of emphasis for investigators to have a multidisciplinary team, to have cross-cutting collaborative teams to answer the questions on inflammation. At this point, I am going to turn my presentation to my colleague, George, who is the program officer for cornea program, as well as for the ocular immunology, inflammation, and infection program. And what she is going to do in the next few slides is walk you through what are some of the questions that we've been thinking in these 3 bins: the normal or the homeostasis state, the disease state, and the resolution. And just in order to avoid any computer glitches, I will go on with the presentation. George will talk, and when she tells me to move on to the next slide, I will do that. So George, I'm moving to the next slide, and which is the normal homeostasis.
Slide, normal homeostasis. 5-stage connected circle diagram. In the center is homeostasis, maintaining balance. Starting from the top circle, what are the resident immune cells in the anterior segment and where do they reside? Next circle, what interactions do resident immune cells have with other cells in the anterior segment? Next circle, how does signaling of non-immune cells contribute to immune homeostasis in the eye? Next circle, how do cells of innate and adaptive immunity contribute to homeostasis in the eye? And lastly, models, human/animal.
Thank you, Sangeeta, for that great background. As Sangeeta mentioned, can everybody hear me okay? As Sangeeta mentioned, I'm going to go over some of the questions that we developed for each of the 3 themes that came from the analysis we did. And for the normal and homeostasis part of the theme, some of the questions are pretty basic, like what are the resident immune cells in the anterior segment and where do they reside? That seems like a pretty basic question, but something we still don't know. What interactions do resident immune cells have with other cells in the anterior segment? And how does signaling of non-immune cells contribute to immune homeostasis in the eye? How do cells of innate and adaptive immunity contribute to homeostasis in the eye? And there's a greater need to have better animal and human models that help us understand if there are any changes in signaling pathways of the immune system and how this affects the phenotypes we see. Next slide.
Slide, disease/visual disorders.
And then the next theme is diseases in visual disorders. And I'm displaying, I should have said this on the last slide, but I slipped my mind. There's a diagram here with an eye in the center and 6 circles around it. And I'm just going to go around each circle one by one and give you an example of a question. There's a need to have better animal models that can recapitulate inflammatory components observed in human conditions. How do inflammasomes influence disease pathogenesis in the anterior segment? What are the changes in neuroimmune crosstalk in normal state that lead to a disease state? There's a greater need to have biomarkers for visual disorders. We need to understand the obstacles for therapies in various diseases and how we can overcome them. And the last question is, what are the changes in the ocular biofilm that take place with the use of ophthalmic devices such as contact lenses and nylon sutures? And how do these changes in the ocular biofilm result in infectious diseases such as keratitis and uveitis? Next slide.
Slide, resolution of inflammation. Diagram, a box divided through the middle into 4 segments. The center is another box which reads, moving beyond corticosteroid. And
the last theme we had was resolution of inflammation. As you all know, resolution of inflammation is a very complex process. Previous research has mainly targeted anti-inflammatories to suppress the initiation and propagation of inflammation. However, much research is needed on how to use agents that resolve inflammation, such as lipid mediators that promote resolution and tissue regeneration. What are the processes by which acute and or chronic eye inflammation resolved? And are there general mechanisms that promote the resolution of inflammation, or are the mechanisms involved specifically related to the eye? There's a greater need for acquisition of human clinical samples to understand the resolution of inflammation. In addition, we need better animal models that can recapitulate inflammatory components observed in human eye diseases. We need to understand whether one drug or a combination of drugs is needed for resolution of inflammation. So overall, we need to move beyond corticosteroids. Next slide.
Slide, possible topics.
So taking all these questions into considerations, we came down to 3 possible overall topics. Dynamic neuroimmune interactions in the transition from normal homeostatic state to disorders, immunosenescence and its role in inflammation, and resolution of inflammation. Next slide.
Slide, inflammation. 3-circle Venn diagram reads RFI, portfolio analysis and program planning. 3 arrows point to workshop, NOSIs, and FOAs.
Our hope is to take all the information gathered from the RFI and the program planning, which is still ongoing, so we expect more ideas from it, and from our portfolio analysis, and have targeted workshops or town halls, NOSIs, and along the way, we'll keep you as council updated and get your input so that we can develop FOAs as we've done in the past. Next slide.
Slide. Thank you.
Thank you. Do you guys have any questions for me or Sangeeta?
So Russ has a question. Russ, why don't you go ahead?
Great, thank you. Thank you, Sangeeta and George, for a terrific summary of this field. It's really interesting to see how this has evolved from the anterior segment initiative that we just talked about in January into a bit of a different emphasis at this point. I want to just highlight one thing that George said at the end. I think the need for town halls here is enormous. This is a bit of a balkanized field as someone who works in the field. The granularity of whether you study corneal immune responses or uveitic immune responses or scleritis or left eye responses or right eye responses is pretty remarkable. And I think the community needs to get together as a whole to really find the common ground that would be best suited for an FOA in terms of moving the field forward. But I really applaud the emphasis on this field. This has been an area that hasn't received a lot of resources of late, and it's a major cause of blindness as well as disability in this country and worldwide. And I very much appreciate the attention to it.
Thank you, Russ. I think that's really a very good point to make. I really appreciate that comment. Yes, thank you.
Annie, you're muted.
Okay, sorry. We have another question from Council Member Mary Ann Stepp. Go ahead, Mary Ann.
Am I unmuted? Can you hear me, guys?
Yes, we can hear you.
I want to thank everybody for what they've done over this topic over the past several months. This is a terrific summation of the major problems that are going on. I'm really impressed that 52% of the issues related to inflammation, that really just targets us to that issue. And Russ is right. We need to be communicating front of the eye, back of the eye, different aspects of this. So I'm really thrilled that you guys have made this much progress. So looking forward to see what happens next.
Thank you.
And then a question from Council Member Eduardo Alfonso.
Thank you, and thank you, Sangeeta and George. Great summary, and Russ, thanks for working in this area. A question and maybe more of an observation than a question, because we may not have the answer. When you look at the number of studies that are listed on the clinicaltrials.gov that are probably funded through industry, and are we able to have a more granular look at that to see how we can somehow partner in this phase or this next phase of planning with industry, and being able to have NEI as part of a industry partially funded clinical trial, because this is an area that I think strategically in the future may lose some impetus as the economy rebounds or not in the next 2 or 3 years, and perhaps those numbers are going to decrease significantly.
I think, Alfonso, you bring a really good point on private public partnership. You know, as you know, for all our clinical trials that whenever we fund them, we bring it to council attention, and there is, you know, partnership with the pharmaceutical companies, whether it's donation of drug or devices or something, we do have them. But the other way around, where we as NIH can do it, I think that's a great point. And maybe, Mike, you want to say something to that thing?
Yeah, so I think this is a really great suggestion, Sangeeta, that we take a deeper look at the topics that have been chosen, you know, particularly by the industry folks for their clinical trials. You know, they generally have the deeper pockets for being able to run the clinical trials. But one NIH's role in most of these things is to identify novel targets for druggable therapies. So if we can maybe find out the kinds of areas that industry folks have been interested in, we could maybe direct our more basic research efforts at trying to find more targets for them to be able to conduct clinical trials on. So it's a great idea.
Yep. I think so too. Very good idea. Thank you, Alfonso.
All right. What I'd like to do, there's no action right now that council has to take on this. This was mainly informational, but thank you for your input. And afterwards, please feel free to email Sangeeta or George with your ideas and to further communicate with them. Right now, though, I'd like to turn this back over to Dr. Taminia, and she can just lead a general council discussion. Anything else that might be on your mind, or if you want to speak further about any of the talks that were presented today, please feel free to do that now.
Thanks, Annie. So first, I'd like to thank all of the presenters for today's presentations. Great job, everybody. I know this virtual atmosphere can be a little bit disconcerting. The other thing I wanted to, person I wanted to thank is Russ. Great job giving us a synopsis of Council of Councils. And I couldn't agree with you more that there needs to be a more concerted effort at coordinated communication between NEI and you and representation on Council of Councils, because there are some very, very powerful hot topics. And as you said, for us to have a voice at the table. So I think that we need to work on that. So I think that's a great idea, and we should do that. So I wanted to ask, since there were no questions about strategic planning, and I know Sheffy presented a lot of information about the process and the information gathering, and you might have had some time between his presentation and now to think about the topics. I wanted to open it up to you to see if you had any questions for him or for anybody else on some of the presentations that you heard.
People could just feel free to unmute themselves, or if you want to raise your hand, I can unmute you or call on you to unmute.
This is Russ. I'll jump in. First, thank you, Santa, for the kind words about the Council of Councils, and I do look forward to opening up some sort of a regular communication channel, which I think would be very valuable. I also want to really applaud Sheffy for the job that he's done in leading these planning sessions. Having been a panelist on the immunology session, they've been really provocative. They get pretty well into the weeds, which is where we need to be on some of this. The one concern I have is that, at least my experience on the immunology panel has been that 3 hours so far and 4.5 hours total is going to be just enough to scratch the surface a little bit on some of these topics. And, you know, as we look to a 5-year plan, everyone's time is quite limited, and I know we don't have infinite resources to do this in great depth, but I think the next stage in the process, which is the distillation and then the editing of these documents and their final distillation down into the report, really needs a lot of attention. I hope that the charge that will go to the committees is very clear in terms of what it's hoped for deliverables, and to make that concrete. It would be really good to have some sense of, you know, how many areas of investigation are within the bandwidth of a strategic plan. You can't do everything well, but you can do many things well. And sort of what's the prioritization that the NEI staff would want from the committees of the many things that have come up. And, you know, you hate to choose among your children, but, you know, as you think about, well, if we have limited resources, should they go 50-50 into microbiome and resident immune cell studies, or is there consensus on the panel that one of those may be a richer vein than the other, or more important to progress in the field? And those are very hard conversations that, at least on our panel, we haven't had yet. But I think they're somewhat essential to making a real working document. And again, I really applaud your efforts on this, but I think the hard work is the next step after the ideas are gathered, to try to distill and prioritize them.
Thanks, Russ. You're right. It's been a lot of, we've gathered a lot of work. This is, as we've called this, the information gathering phase. We've gathered a lot of information. Our RFA had 250 some odd responses, 252 responses, and that led to the panels. And as you know, it's not just the 3 hour, 4.5 hour meetings that we are having, but we also collected a lot of written materials before some of the calls. So we have, I think, something in the order of 30 to 40 pages per panel of notes and ideas. The work ahead of us is a little bit daunting in trying to distill that into something. And so we are going to try to do the lion's share of lifting at the staff level, trying to distill what we can, but then of course feed this back to the chairs and to the panels to really make sure that we haven't missed some key things in that process. But I want to point out that there's really, we're trying to get 2 for the price of 1 in this whole process. So we have a public facing plan. This was council's idea. In fact, I believe, Russ, it was your idea at council to really have the public facing document is typically going to be, it's on the order of a 50-page document. That's something that we can present to Congress, and they can use as messaging. And these are the high top level things that NEI sees as priorities going forward. But as long as we've had these groups together, we've really gotten into the weeds, and a lot of these weeds are not going to fit into a public-facing document. We're also developing, and we're calling it the implementation document, but it's really has a lot more of the details and things that didn't quite that are not going to make into the public-facing document are still things that we want to capture and provide as input to our staff and to our new director. And so right now it's following the same format as the same template that we have for the public document, but with a lot more details. But also within that, we have been asking some of the panels to come up, set the priorities, figure out where do you rank these things. I know the public health panel used a, I can't remember what the analysis, they really tried to really drill down at different levels from their top priorities. And some of the data science panel is using a Delphi method to really come up with priorities right now. So we are asking the panels to set priorities either during the panel meetings themselves or as we take it to the writing phase. So what you are saying is the case that sometimes there's so much detail, so many things that we just can't include. We are trying our best to make sure that we have key staff that are sitting in on the calls so we can capture that as we take it at the programmatic level, but also that we're developing this whole document, this parallel implementation document that will really have a lot more of the details that might not appear in the public-facing document.
That's great, Sheffy. The one other thought that comes from this is, in addition to a 2-for-1, maybe there's a 3-for-1 here. Would it be worth offering the chairs of the various panels the opportunity to write this up as a separate white paper of the state of computational ophthalmology or artificial intelligence or any of these areas, and look to potentially publish these in the peer-reviewed literature. I don't know if Marco Zarbin is on the call today, but for some of these, it would seem like a natural fit in a journal like TVST or IOVS or something like that. But it would allow a depth to the discussion that isn't going to be in the public-facing document and might not be available anywhere. And I'd hate to see the deliberations of these committees not be fully mined for what's in them. I think there's a lot of great discussion coming out of these, and we can really make some, have some documentation that would help drive this forward and also provide reassurance to the scientific community that these issues were discussed in depth before the priorities were established.
I like that. I think there's also opportunity there's some of these are relatively new areas for NEI, particularly how we're thinking about public health and data science. And so those are particularly areas that are potentially ripe for publication and how we're approaching them. But that's a good idea, Russ.
Thank you, Russ. Anyone else have any questions about the strategic plan and the process? One other thing that I wanted to mention was that
I'm sorry, Santa, I think Carol was trying to speak, but she was muted.
I was muted.
Okay, sure, Carol, go ahead.
No, I just wanted to echo what Russ said about doing a more timely white paper for some of these, for all of the committees, because there's a lot going on in science right now, and different from whenever the last strategic plan came out, which was more of a descriptive, almost historical document. I would welcome a short papers for each of these committees on where we are and what needs to be done, or what are the questions going forward. Especially with the first three, I'm very interested in, and also the one on neuroimmunology. So I would encourage Shefa and whoever, the various chairs of the committee to think about doing this in a more timely way.
And you're right, Carol. I mean, this has been a much more a forward-looking kind of exercise, and we really thank everyone enough for the enthusiasm and engagement we've had in this process. I feel like I've been with NEI for like 100 years, and this has really been a very engaged strategic planning process, and that's a credit to everybody who's on the panels and have been working very hard to really accomplish something worthwhile for us. So it's really fabulous, and we shouldn't let that go to waste.
I actually would like to refer, sorry, can you hear me?
Yes.
Sorry. I would like to refer everyone to the new document, the new BRAIN initiative, BRAIN 2025 version 2, which went through a similar process, much more involved town meetings, just to see what was done with that. That's a very long document, but as an example.
And it looks like Mary Ann Stepp has a question too. So Mary Ann, do you want to?
I think that this planning process you guys have gone through is really going to put us in a terrific position with the new chairman coming in. I'm sure they're going to really appreciate all of the work that has been done. They're going to have an updated, plan for going forward, and it's really going to jumpstart, what happens in the next year. So I think it really makes me feel good knowing that in these uncertain times, you guys have been working so hard at making sure that the future is going to work out well for all of us. So I just wanted to say that. I appreciate what you guys have been doing.
Thank you, Mary Ann.
As much as I'm thankful that we could meet virtually, I know that this venue isn't as conducive to discussions and a lot of feedback. But please, any council member who, if you have any concerns, anything else you want to say, now is the time. So please feel free to do that.
This is Katia. I just wanted to ask Russ if he could talk a little bit more about the programs for the Centers of Excellence. I was interested on that.
Centers of Excellence. I think from the Council of Councils review, is that the reference?
Yes, please.
So it wasn't necessarily. Oh, I see. So for the goal is to try to improve the success of underrepresented groups in science, and a blue ribbon panel met to review progress in this domain. And one of their conclusions was that although many individual institutions have succeeded in recruiting faculty from historically underrepresented communities, the success rate has not been necessarily what NIH would like to see in that domain. When they did sort of a root cause analysis, there was some consensus that perhaps part of this was that you had many, many programs recruiting relatively few underrepresented faculty each, and that the support structure within any given institution was insufficient to really help foster their careers. And in particular, peer mentoring is a real challenge if there are few peers who have been successful. So the initiative was really designed to allow a competitive process to create centers of excellence at a number of universities through the universities and other grantee institutions throughout the country that would have a goal with funds available from this program to recruit outstanding underrepresented minority faculty and provide a local, deep support network of other successful faculty, as well as sort of a career development plan for recruited faculty that would be specific to that institution. I think there was a lot of enthusiasm for that idea of creating an infrastructure that promoted success. The discussion that countered that a bit was that effectively this would create a handful, perhaps a dozen or so institutions that had tremendous resources to build a community of scientists from underrepresented backgrounds, and that could have unintended consequences, one of them being recruitment of such faculty away from existing institutions, because all other things being equal, it sounds like it would be better to be in a place like that, and that could counter efforts from other institutions that they have already made, and in fact reduce diversity at many institutions. And the second was just the issue of concentrating quite a lot of resources into a small number of institutions. I think the total proposed price tag on this program was somewhere around, over 5 years, $250 million. And the proposal was somewhere in the neighborhood of about a dozen to 2 dozen programs. And so that's a lot of resources being concentrated in a relatively small number of programs, which might have, again, unintended consequences by reducing support for diversity hiring at institutions that didn't have one of these grants. At the end of that discussion, which was pretty robust and really many stakeholders weighed in, the Council of Councils did provide concept clearance and voted for concept clearance around this, this idea. I have not seen that there's an FOA at this point. It would be pretty soon, I think, for that to be out. But I suspect that this is moving forward at this point. Does that provide the information you wanted, Katja?
Yes, yes. I was just concerned that it was going to be focused, as you said, to institutions that already have a lot of, funding, and the ones that didn't were going to be just not. I mean, I don't know what was the focus on big institutions versus on represented institutions as well.
Yeah, I think there was quite a bit of discussion about the selection process, and to ensure that this wasn't a winner take all process where institutions that have deep pockets and big resources and can support this type of program would have more. I think there was quite a bit of discussion about ensuring a large diversity of different types of institutions as eventually recipients of these awards. So that was discussed.
And if this FOA comes out, it's going to be from what program?
It'll be a common fund program, so it will come out from the director's office as a common fund initiative. I don't think that this will be administered by individual institutes. So there won't, my understanding is there won't be an NEI initiative for diversity in hiring. This will be really at the director's level. Santa, you can correct me if I'm mistaken, but I think that was the impression I got.
No, that's correct, Russ. You're right. There was also conversation, Katja, at the leadership forum about, just exactly what you're worried about. The larger institutions who may be already well-funded and well-poised, getting more money, and those other institutions who are trying their best but don't have funding to put good programs in place being left out. So there was a lot of conversations about how to get those already underfunded institutions up so that they could stand up good, meaningful programs, which might even be more successful than some of the larger institutions. So there's definitely awareness of those issues. There is also conversation about why there seems to be, in some instances, not folks from underrepresented backgrounds not getting high priority scores. And it has to do on when we're looking back at the types of applications they put in, they tend to be interested in different things than what we consider, the normal traditional scientific pathways. And so they're more interested in social science and behavioral science and community science, and those tend to not score as well in study sections. So there is some talk about, what do we do about that and how do we address that? Because those are definitely issues that are important, and they're important to our underrepresented populations. So how do we make them more successful in the review process? So these are definitely topics that we're discussing at the leadership level, and they're still ongoing because obviously we don't have a perfect answer.
Okay, thank you very much for both.
Is there any other feedback, any other questions or concerns from council members? Do any of our panelists want to weigh in on any of the topics? And Mary Elizabeth, why don't you ask your question?
So I also have a question regarding the Council of Councils. And Russ, thank you for your leadership there. And Santa, thank you for your input. You had mentioned that that NEI is about, I believe you said about 2%. And so sometimes it's hard to get our voice heard. And that made me wonder if there were other institutes that were also, not highly represented, but were able to get their voice heard. And if there were strategies that we could learn from them, or whether how much of a problem, I guess, this is about trying to get NEI's sort of voice heard within this council. Maybe I misunderstood.
No, Emmy, I appreciate your point, and it's exactly right. NIH is really a huge institution, and sitting on this Council of Councils, you get a sense for the breadth of this organization. The budget, I think Karen said this year, is $38 billion a year. And if you think about corporate America, the number of companies that have revenues in the range of $38 billion a year puts you well into the Fortune 100, if not the Fortune 50. So, if you want to think about it that way, NIH is really, a giant on the size of a Boeing or a Microsoft or something like that in terms of its annual expenditures. And we are less than $1 billion of that. We're $800 million of that 40 billion. So we're a relatively small institute, and there is, even around the table at Council of Councils, I think, a lot of attention paid to program domains that fall within some of the larger institutes, whether that's NCI or allergy and infectious disease, or some of the other larger institutes, even neurologic disease and stroke. I think the key is to utilize that forum effectively and speak up when there is an issue that impacts us, to make sure that we get some recognition and can be part of some of these programs where we have something unique to contribute. As an example, as I think Shefa alluded to, in the area of data science, really, we have a leadership position. The first FDA-approved deep learning system is for the diagnosis of diabetic retinopathy. And in the areas of gene therapy and regenerative medicine, we have a leadership position despite being only 2% of the NIH budget. So those are areas where it's fairly easy for us to say, we should contribute to this. And as an example, my hope is that perhaps we can have a significant role in some of the discussions around big data format, which we discussed at the May Council of Councils meeting. How do we create data sets and compliant data sets so that everyone can use them? That's a really important question. NEI shouldn't be paying 3 times for the same study because people don't release their data sets, or they're available in an unusable form. So what I would like to do, as I mentioned, is really have an open channel of communication to this council, presumably through the DEA, the Director of Extramural Activities. And before each Council of Councils, just review the upcoming agenda and the pre-materials and just see if this council has insights or input that it would like communicated to the larger table. It's actually a little hard to get speaking time at Council of Councils. People are not shy about speaking up, and it's not unusual for 10 councilors to speak on everything that comes up. So we get our 30 to 60 seconds to say our piece, which means we can't comment on everything. And that's where I'd like this council to really provide some help for me to figure out when to speak up and what to prioritize to ensure that we get our voice heard.
Great, thank you, Russ.
I also see Bruce Reed's hand up. So Bruce?
Thank you. Santa, I wanted to follow up on your remarks. You were talking about the observation that there are differences in the award rates that the different scientific topics have, and that the topics that tend to be favored by minorities tend to have lower award rates. And the thing that I want to follow up on and try to clarify for people is that the quick conclusion on this is that these topics do worse because reviewers don't like them. And the picture is and that's the argument that was presented in the paper that many people are familiar with, Hoppe paper in Science Advances. I just want to get people to think that the picture is more complicated than that. And so if I can just take a second, I'll try to be concise here. But as I say, the observation is that if you divide science into different topics, those, and then you look at the award rates for applications in those different topic areas, they vary considerably. They vary from 7% to 29% success. And the topics that African American applicants tend to favor fall into the ones where the award rates are lower. It's also true that each IC has its own favorite topics. So NEI, for example, doesn't fund a lot of implementation, community health kinds of studies. And if you look at award rates across institutes, they also vary tremendously, right? So if you look at the institutes that support work that tends to be favored by African American Black PIs, those institutes tend to have very low award rates. If you just look across NIH, the award rates for different institutes also range tremendously. They range from about 9% up to 29%. So as I say, the award rates for the institutes that support this kind of science are really low. They're at the low end of NIH. Whereas the award rates for the institutes that support work that is not so favored by minority investigators tend to be very high. So for example, as I say, the NEI doesn't support a lot of work in that area, and your rates are at the top, really, of the NIH distribution. So here's my bottom line point that I want to make. People make the quick equation of differences in award rates to bias in review. And the picture is substantially more complex than that. And one of the things that I would encourage people to think about is the contribution of institute funding rates and how that plays out in this picture.
Thanks for your comments, Bruce.
Yeah, and Russ, you had your hand up again?
Yeah, I just want to address that just a bit. And Bruce, I appreciate the complexities of this, and clearly this is a very, very complex issue. But I do think that the NEI has paid a lot of attention to issues of healthcare disparity and issues of healthcare delivery. And in particular, one of the largest grants in this portfolio of the Institute, clinical grants in the last decade, is the Latino eye disease study, which is an enormous multi-center study looking at healthcare disparities, specifically in the Latino community, that have particular eye problems present at much higher prevalence than other communities. So I think broad brush strokes saying that, you know, you do or don't like funding initiatives in these domains have to be very careful. I mean, the devil is in the details of what's being funded. But if the impression outside NEI is that NEI does not have appetite for looking at healthcare disparity issues or other issues that traditionally attract underrepresented scientists, as a counselor and as someone who signed off on the billet of funded grants, that's not my impression at all. And I think NEI does pay a great deal of attention to areas of healthcare disparity.
Yeah, if I can respond. So the issue isn't so much whether you support any work in that. It's more understanding this broader picture that's been pushed out. And so if you look at who supports the scientific topics that are favored by Black and African American investigators, most of that work is supported by institutes like Minority Health, Nursing. And if you look at their award rates, they're very low. So that's the basic point that I want to make. And you know, I understand that NEI certainly has an interest in some of these topics, but if you look at where the bulk of this work is supported, it's in other institutes, and their award rates are not nearly as strong as yours.
Okay. Any other input from council members? Not seeing anything, I think we could conclude our open session. I want to thank our interpreters and our cart rider, whose hand must be ready to fall off at this point. I'd also like to thank Michael Wright, our IT person who really did the lion's share in putting together this Zoom webinar. I thank him very much for that. And what I'd like to do at this point is take about an hour lunch break. I don't know if I'll take your input if you want to go a little bit shorter than that. We don't have a long closed session this afternoon. So, but I don't know how long a break people would like to have. So why don't you just speak up? We can take as a full hour. We can go a little bit shorter, maybe just a 45-minute break. What's your feeling about that?
I'd rather do shorter so that we can get done earlier.
Okay.
30 to 45 minutes is plenty.
Okay.
Second that.
Right.
That's fine.
Then why don't we take about a 40-minute break, and that would put us back here at about 2:10 p.m. Eastern time. Would that be acceptable? Yeah.
Yes.
Okay. All right.
Yep.
All right. And then if any council member would like me, I can just resend the Zoom link to the closed session again, if you'd like to have that. Maybe I'll just do that so you've got it. Okay.
That'd be great, Annie. Thank you.
All right. All right. So see you back at 2:10. Thank you, everyone. Thank you, attendees. Thank you, panelists. Thank you for our closed caption and interpretive help very much.